All right. Hi, everybody. My name is Ann Gill. I am going to be with y'all for the first 2.5 hours of this CRAM session. And then I will be getting off, but I'm going to be doing the first 4 units with you. So a little bit about myself is I come from Texas. I am right outside of Houston in Cypress. I teach at Cypher High School, and I hope I have my students that are watching, and I hope all of you that are around the nation tonight are getting ready, and you're going to be so prepared for the AP test tomorrow. I do want to go ahead and share my screen with y'all. Window. No, I need to do the Chrome tab. I think that's what I need to do. We'll see if I did that right. Hopefully I did. I am going to go ahead and share my screen and put it in present mode. All right. I hope y'all can see that okay. Definitely let me know, Peter or Jed, if I'm not okay. So let's go to the slides. Awesome. So we are going to, Amanda's actually going to be coming on in just a little bit to talk to y'all. So I am going to skip over that because she'll be coming in just a little bit to talk. So let's go here. It is being recorded tonight, and so I did want to go ahead and put that out there. And this might actually appear later on Fiveable or possibly YouTube, other places. So if you would, if you have a question and you do put it up, I know that Amanda is also going to tell you, and I think she's actually ready. So I'm going to let her take it over. Amanda is here. She is the co-founder of Fiveable, and she would like to talk to y'all for a few minutes.
What's up, AP Bio? It's Amanda here. I'm so excited to come and chat with you. It's really me. I'm live from the Milwaukee headquarters where Fiveable is located. So I wanted to pop in to tell you a couple quick things that are just kind of exciting. First, you're in the right place tonight to prepare for bio tomorrow. So you're going to be covering a lot of content and everything you need to know for the test. If you have any questions on content or FRQs or anything exam related, make sure to post that in the discussion thread so that our TAs can help you out. And if you have tech issues or anything Fiveable related, post those in the chat box with my little mustache. The student success team will help you there. The other cool thing is that we have a new part of Fiveable now called Hours, where you can create study groups with other friends. And so as you're studying, if you want to like set up an Hours group, you can actually have like a little to-do list where you keep track of the things that you want to make sure to cover and check those off as you go and keep each other accountable. And then lastly, we are doing our annual social media contest. So we've already given away several different prizes. We're giving away $100 every day. So we've already been doing this for the last like 2 weeks. And tonight we're giving away another $100 for a student that posts on either Twitter or Instagram or TikTok about your AP experience. So it could be something about, your study space, something biology related, something calling out College Board's nonsense. We love that. Something fun. So post it, use the hashtag #YesWeCram, and make sure to tag us at ThinkFiveable so that we see it. And our student interns are going to be choosing the best of the best and sending $100 to someone. So that basically pays for your AP test. So if you're watching this as a replay, that is still happening. So make sure to pop in at some point to play. And then very, very lastly, I just wanted to let you all know that I am so proud of you. I know that this has been a wild year. You've made it through a lot. So the stress levels are very high, when we're thinking about the exam. We know the stress is there. So just please take a deep breath, just relax for 1 second and just say it out loud. I got this. Like the one thing that's going to matter the most on the test tomorrow is that you are confident, right? That you just are like, I'm gonna be fine. I'm gonna do this. I got everything I need in my head. I'm in this cram session right now. I've done everything I can. This year has been a lot, and I made it through. I made it to the end, and now I'm gonna show what I can do. And worst case scenario, you get a 1, but who cares? You made it through this year, and you showed how resilient you are. You've done the work. So we are incredibly proud of you for all that you've done. Just know that we're here for you for anything that you need, whether that's AP related or you just need someone to talk to, you're getting really stressed out. We got you. We got your back. Okay. So let us know if you need anything. We'll be in the Discord chatting and, answering questions all night. So I'll send it back to you and have fun tonight reviewing all of AP Bio. Bye.
Definitely, I'm going to share my screen again. Make sure I'm going to put that up in just a second so that y'all can see what Amanda said about what to go. Sorry, I have a hard time sometimes talking about different things. So this is what she said right here about yes, we cram and with Fiveable and then tag at think Fiveable. Do it on any of those places and they are going to be it out and somebody's going to win. Maybe it can be you. And I can't emphasize enough what Amanda said before we even get started. And that is that let's take a deep breath. Let's think about what's going on. And y'all have prepared. Y'all are ready. Y'all are here. I mean, good gracious, that is dedication. You are here for this, and you are here to come and learn more. Hopefully, not a lot. Hopefully, it's just coming back and going, oh, I remember that. And being able to know when you walk in tomorrow to the test that you feel good about it. Take yourself a deep breath, just like she said, and we got it. So with that, let's get to the first little part. The first little part I want to talk to you about, and this is on College Board's website, but of course, y'all have access to this PowerPoint at underneath. Excuse me. And you can most definitely look at these later. You might want to just scan over them if you have not looked at them. These are the task verbs because, as we know, we have two parts to the AP test. We have the multiple choice, which you will get an hour and a half, and you will then have an hour and a half to do the FRQs. With the FRQs, these are the task verbs that College Board has said that they're going to use for y'all. I hope tonight that Carrie and I can get to where we can do some FRQs with you. I'm watching my time though, so I will see where I can and can't put those in. And these are the rest of the task verbs. So for sure, I don't want to spend a lot of time sitting and going over those because I want to get to content. But this is something for y'all to look at if you have just a few minutes just to kind of glance at. With the free response, there are, this is the first year we're going to see this, there are going to be 6 of them. With the 6 FRQs, you're going to get to see that there will be 2 long FRQs, and then there will be 4 short FRQs. With the long FRQs, they are telling us that they should be 8 to 10 points. Hopefully you've done some FRQs in class and you've got to see some long FRQs. But if you haven't, there are some embedded in this PowerPoint tonight with the rubrics, so you would be able to look at them if you needed to before you went to bed. With the first 2, these are, they say 8 to 10. Everyone I've seen so far has been 10. No, it's been 9 or 10. But what they're saying is there will be 4 parts to them. They are telling you that the first one, you are going to be given some kind of scenario. You're going to be given a data table, and you're going to be possibly given a graph. Now, you will have to analyze what you see there. And y'all, and I've told my students this, if you are given a data chart and you are given, or you're giving a graph, and you are asked to analyze, you see how on part B it says there, the first one, justify predictions. Y'all, they don't want you coming that out of thin air. They don't want you just making up something, pulling it out of the air. They want you using the information that they have given you in the chart and in the graph to be able to justify your answer. That is so very important. So if they give you a graph and they ask you to, okay, analyze, so maybe you need to say that there is a direct relationship. Maybe you have to find the slope. Maybe you are going to be talking about how things compare with the error bars, but you are going to have to analyze that. Then they want you to justify what you just analyzed, and you will be using the information that's there. How that's different in the second question is that in the second question, you will have to make a graph. Now, if I'm a quick side note, if you are one of the ones who is not taking the AP test tomorrow and you are actually going to be taking a digital one, excuse me, the digital AP test will not have you making a graph. You instead will have to analyze again. You will be given information and you'll have to give some of the experimental procedures. You'll have to give some more information, but you won't actually, because they don't want anybody doing it digital to have to draw and submit. Now, all of us taking one tomorrow, we will see a graph. We will see confidence intervals. They're straight out telling us that. So I know I've worked with my kids on this, that you've got to make sure you know how to do those error bars. And we are going to talk a little bit about that tonight. So that's the first two. Then we've got the next two. These are both, now the rest of them are going to be 6 short ones is what they call them. And so they are all four, I said 6, I'm so sorry, 4 short ones. And they are all 4 points apiece. Okay, now with this, there are different things that they're saying. So if you'll notice on number 3, they're saying that they're going to give you a lab investigation scenario. It's okay if you have not done that lab. They're going to give you some information about that lab, and they're going to give you the information you need to then be able, based on the content that we've learned, to be able to then analyze that particular lab. The next question, you're now going to be given a scenario. So now you're going back to, maybe it is about a disease. Maybe it is about a pedigree. Maybe it is about a disorder, and this disorder is causing this mutate, or this mutation has caused this disorder, and then they go in. But the thing there is, you'll notice disruption. Oh my heavens! In my mind, I think about enzymes, and I think about how a protein can become misshapen. And if it becomes misshaped, how then, what molecularly that means. And so they're going to give you information. There could be a picture there. If ever, y'all, there is a picture, a graph, or a chart, they didn't do it for fun. You need to use it somehow. So that's for the first 4 that we've got so far. And then for the last 2, you're now going to be looking at, and this one they tell you for sure, there will be a model or a visual representation, and it's going to accompany a scenario. And in here, now you're going to have to talk about how that relates. And you'll notice they use these task verbs. They go ahead and tell you that they're looking at that you're going to have to describe. You're going to have to explain. There could be in there also, they could ask you about a null hypothesis for sure, because if you'll notice on the very last one, this is where they're going to give you, with a data in a graph, a table, or possibly something else. They're then going to be assessing it, looking at the data, and you're going to have to come up with hypothesis, possibly a prediction. It could be a null hypothesis. So between Carrie and I tonight, we will be talking about that, so that y'all can talk about, because maybe you haven't heard about experimental design enough. Maybe you need a little bit of assistance on that. We are going to talk about that tonight. So let me check this real quick. Yes, if you will tell me how to turn my captions on. You are so right. I knew I needed to do that. Do I need to stop sharing my screen? I apologize, everybody. I am so crazy and I'm new and I don't know everything. Do I need to stop sharing? I will go to the next one though while I'm waiting. Because we're going to start with Unit 1. I knew, Carrie even told me, you need to make sure you have your captions on. She always forgets. Yes. Okay, I am going to stop my sharing for a second because I have a feeling that I need to I apologize, everyone. No. Control Shift C. Oh, okay. So I can do it while I'm actually sharing my screen. Gonna do it again, folks, while you're presenting. Thank you. I don't know where I would be without these people because they are incredible. Control Shift C. Allow. All right. Okay, we're going to start with Unit 1. With Unit 1, there is, if we talk about in the breakdown, College Board gives us a breakdown, and they tell us that in Unit 1, which is the chemistry of life, we should see of the entire AP test. So we're talking about from the multiple choice, which, by the way, there are 60 multiple choice questions. Of those 60 multiple choice questions, you could be asked to do math, and I would expect that there will be some math. Hopefully this is not brand new tonight and that you have heard stuff, but I do want to emphasize, so there will very well be math in the multiple choice. There could be math in the FRQ section. You will be given a formula chart for both sections. There will be a booklet for the multiple choice. There will be a booklet for the FRQs, and there will be a formula chart at the beginning of both of those. So you do not have to worry about having those or anything like that. It will be there. You do just need to make sure that you have a calculator with you because if there is math, they assume that you have a calculator to be able to do that. In the chemistry of life, they tell us that it is 8 to 11% of the total test will come from that. Well, what is the chemistry of life? It is the 4 macromolecules. And this is not like just a possibility. The biggest one that they love to talk about, of course, are the proteins. But that does not mean that they will not hit on the other ones. And I would expect there to be some kind of question pertaining to each one of those. When we think about but they like to, because I've been a reader now for, this will be my 10th summer doing it. When we're at the reading, it's interesting. We get to talk about multiple choice as well. They really like to put two things together. So they could be talking about the cell membrane. And so they're talking about the structural setup of the cell membrane, which is the next unit. But then they go in and they're talking about the lipids, and they're talking maybe about the cholesterol, and how the cholesterol adds some of the rigid components to the cell membrane. Or they are talking about how, because the phospholipid bilayer has the hydrophilic middle portions and the hydrophobic outer portions, that causes a problem, because that means that something that's hydrophilic is actually going to be repelled, and that you then need to have something that is going to be able to have a channel to go through to be able to get inside the cell or outside of the cell, if that's the case. Now, with in the sense of the monomer and the polymer, On old tests, I have seen the structural components, and so I'm going to kind of start moving ahead so that you can see some of the other things because I've got some pictures in here. And so I'm going to come back to that in just a second. I want to show you so with carbohydrates, we normally see that the sugars are going to be rings, and these rings are then put together, and those can then be larger, and so we can have the monomer form, which is a monosaccharide, all the way up to like a huge one, which is cellulose, that our body cannot digest. And so when talking about that, you'll see though that it's very much the same idea. It's this linear type structure that then can branch out and become sheets, or excuse me, can branch out and become very much like a tree. Whereas if we look at lipids, our lipids have a very much different component to them. And that is because of the fact that they have two parts. They have they have no monomer or polymer in that respect. They are just one thing, that monomer, and that is considered that it is a glycerol and at least one fatty acid. There has to be at least one fatty acid chain. And the fatty acid chain is what comes off of the glycerol. The glycerol is this main, on this picture here, that is the triglyceride, that you then have all these other parts that are these long chains of hydrocarbons. One thing I haven't seen, that was what I was getting at, is that I haven't seen them showing us where there's lots of pictures of having to decipher between what is a lipid, what is a carbohydrate, what is a protein. But I would be expecting that you should know how to do that. And the reason why is it helps with understanding how they work then. So if we go back for a second, dehydration synthesis and hydrolysis, these are the two main types of reactions that happen in our body to be able to put things together or take things apart. So when we're talking about putting things together, that is going to be dehydration synthesis. Dehydration synthesis is where we take the monomers and put them together to make them the polymer. And the polymer then is going to be what we also call a macromolecule. So like for our proteins, it would be a chain of amino acids would be the polymer, which we call a polypeptide chain, or we call it specifically just a protein. But when we're looking at the process here, this is what helps to make our covalent bonds. And these are the things that help us to, like in this next picture, put those monosaccharides together to actually then be turned into a sugar. Now, sugars are made of just carbons, hydrogens, and oxygens, and that's all we see for the carbohydrates. Now, the same thing is true with lipids, it's just not in the same ratio. When we're looking at a lipid, one thing that we really need to think about with a lipid is that there is always going to be these hydrophobic tails and the hydrophilic heads. And I think I said that backwards just a few minutes ago. Huh, I think I said it the other way, and I apologize. So when we're talking about that hydrophilic head is going to be made of that glycerol molecule, that 3-carbon molecule. Now, to be a phospholipid, you actually have some other parts to it. So if you notice this second picture here that has, and I'm not sure if y'all can see my mouse or not, but this second picture that's on the bottom there on the left-hand side, you see that there is this yellow circle, and that's that's the actual phosphate. But then you've got some additional portions on top of it that makes this whole portion attached to the glycerol that turns it into the phospholipid. So when we're talking about a phospholipid, that of course is what helps to make our cell membrane, but they are for sure a lipid. And with this, they are not they're not saturated. They are unsaturated, which is really important. If by chance you were asked something about the fact that a cell membrane being unsaturated is really awesome because it doesn't then, or hopefully won't, solidify. Because something that is going to be saturated, like the top picture, that is a triglyceride that is saturated. There is no double bonds. And if there are no double bonds, then there is a higher chance of it solidifying at lower temperatures. All right, let's go to with the I had mentioned about this. So, like in that, it shows that cholesterol stuck in there, and then I just lost my train of thought. Sorry. So we have the nonpolar portion that are then with those polar portions, and that's what causes problems. If you want to call it problems, and that is the fact that if they have their two parts, if you have something that's trying to get in, such as like it shows in the over here in the writing, such as water, well, that's going to be a problem because water, yes, can slip through, but for the most part, water needs to go through an aquaporin, which is a channel, because of the fact that it has a polarity. So something that has a polarity is not going, or has a charge, is not going to be able to just slip through the membrane, and that could be a problem. But that whole idea of that's where channels come in, and those are all made of proteins, that they are then going to help to be able to allow mass movement, or if let's say it was in a neuron, it would be able to help with the sodium potassiums moving back and forth, but because they both have charges and they cannot just slip through the cell membrane. All right, so now let's talk about, the big guy. Let's talk about proteins for a second. Proteins. Well, the monomer form of a protein is one amino acid. Now, an amino acid is made up of 4 components. And yes, you are expected to know that. 100% you are expected to know about an amino acid. You are expected to know that it has an amino group right here. You are expected to know that it has what is called a carboxyl group to the right here. It has always a hydrogen coming off, and then it has this famous R group. R represents variable, and it can vary, and it does vary. There are 20 amino acids that are found in nature. We as humans need all 20. Our body does not make all 20. Our body only makes 12 of them. So that means that 8 of them we actually have to get from our environment. But regardless of that, of these 20 amino acids, every single amino acid is set up exactly the same. It has one difference every time, and that is that it has a different R group, meaning something different is coming off of that one carbon, off that one part of the carbon. And you'll notice in this picture, they just showed R. They did not show in both of these pictures, they did not show us a different one. It could be as simple as just an H coming off. It could be as complex as a full ring with a sulfur bridge there coming off. But regardless of the fact, it does have all of those different parts. Now, one thing that is special about proteins is that proteins have sulfur, but they do not have phosphorus. There is no phosphorus found in proteins. Whereas then when we talk about nucleic acids in a little bit, in nucleic acids, there is no sulfur, but there is phosphorus. And so that is something that's very unique about the two, and also something that's very special. It helps in also discerning between the two. If you were ever asked anything, is about the fact of what they do have, because carbs and lipids all have carbons, hydrogens, and oxygens. But then proteins and nucleic acids have carbons, hydrogens, and oxygens, of course. But something that is special about them is about the sulfur and the phosphorus. Now, when any of these form bonds, so if I were to go back and talk about carbohydrates bonding together, they do it through dehydration synthesis, and they form a covalent bond. That's extremely important to know, that they form an covalent bond. Covalent bonds in our body are the strongest. I know if you understand chemistry and you are in Chem 2 and things like that, you know about that hydrogen bonds are really strong. Not in our body, though. In our body, hydrogen bonds are extremely weak. So when we're talking about forming proteins, we're going to form proteins or form any macromolecule with covalent bonds. So when the lipids, the glycerols attach to the fatty acids, they do that through covalent bonds. When they do that with the carbohydrates, they do glycosidic linkages, and they do that as a covalent bond. So when proteins bond together, and of course that happens at a ribosome, which Carrie will be talking to you about that. When that happens at a ribosome, that is going to be a covalent bond, but specifically a peptide bond. Now, the function of proteins, 100% through and through, is that they do work. They do so much for us. They are over 50% of our dry mass in our body is actually proteins. It's understandable why when they were searching for DNA, was it DNA or protein that was the genetic material? They lean towards proteins because proteins make up a lot of our body mass. So if we go on and let's talk about proteins, because I feel that this is probably one of the hardest parts because it comes up throughout the entire year. We talk about protein and we talk about what happens when proteins get messed up. When proteins are first made, they are made in the primary structure. The primary structure is going to literally be amino acid bonded to an amino acid and bonded to amino acid and so on. That is exactly what we see. But when we get to the secondary structure is only going to be hydrogen bonding, and it is going to be with the peptides, but the peptide of the backbones. So it's going to be those hydrogens that are coming off. So they're not strong, but what it does is think about if you have a really long string, you need to make it as compact as possible. This is the first way to make it compact. And so it does what are called alpha helixes, so swirls, and beta sheets. And that's like if you took a piece of paper and you folded it on top of itself, that's how it would look, like a pleated sheet, or like if you were making a fan with a piece of paper. That is the secondary. That is not a functional protein. It will not function at just the secondary level. It has to get to the tertiary structure. The tertiary structure is going to be the quote unquote final. A protein can now function at this point. This is going to be, and this is what's so important, how shape determines the function. If the sequence of amino acids gets out of order, or there's a mutation, or a new one comes in, or like one gets put in and it makes a frame shift mutation, or vice versa, we take one out and it makes a frame shift mutation. That changes the entire shape, possibly, of the protein. And if it changes the shape of a protein, it's not going to be able to do its job. Now, not all proteins are enzymes. We have proteins that are all over our body, but enzymes are a subsidiary of proteins. So meaning every single enzyme that exists, and they normally have an ASE ending, they are going to all be proteins, which means they are all made this way. And if they are all made this way, and they get the big word always to use, denatured. If they get denatured, an enzyme will no longer function. And it might be that it can kind of function if it's just one part and the active site is still kind of exposed. But if it gets to the point where it has been denatured, now it's going to start to slow down the rate of a reaction, because that's the whole point of an enzyme, is to help speed it up. And it is then going to possibly stop things from happening. Now, quaternary, which is the fourth level, that is not for everything. That is, some proteins are completely fine going to the third level, and that's it, because they're only made of one strand. If they are made of multiple strands, hemoglobin is a wonderful example, and that's kind of what that picture is right there. Or if you were talking about collagen, those are really two awesome ones to talk about, and they both they have multiple strands. That's only if we have multiple strands do you go to the quaternary. But do you have to have that? No, you do not. Every protein must go through the first, the second, and the third level. Okay. So now if we talked about, and this kind of goes into the idea that, it's not switching, there we go. Globular, yes, that's really the name of it, is they are globs. And so when enzymes, because like I said, all enzymes are a type of protein, they're going to help to catalyze. They're going to help lower that activation energy. Oh, I'm going to go to the next slide because that is something that's really important. If you ever hear or see something about it's lowering the activation energy. So that red line is showing us without the enzyme, and the blue is showing us with the enzyme. And how that's important and why that's important is because of the fact that there are things in our body that wouldn't happen. It would take just too much energy for them to go through to completion, that without an enzyme, they wouldn't work. So an enzyme lowers that energy, and then it's able to do work. The thing about, of course, an enzyme is it doesn't change. It is reusable. It is not used up, and it can, unless becomes denatured or broken down, it is able to be used again. So if I go back over here, what this picture was trying to show us is that that's what happens when things get denatured. They're no longer in that globular shape, but the globular shape is important because what it does is it has a specific shape, which means it has an active site, which we're going to get to that picture in just a second. And that's where the substrate is going to be able to be to fit. I know, why can't we just say reactant? We can't say reactant because we're talking about organic molecules. And if we're talking about organic molecules, we really should use the term substrate. Now, as a reader on for College Board, if we're talking about something and you called it a reactant, we're still going to give you credit. But substrate is the terminology that you really should be using. Okay, so these are because of the fact that you could be given some kind of scenario, and that is a disruption. And in this disruption, they then talk about the enzyme being denatured. Or instead of that, they just give you a graph and they show you that, if you'll notice in that top picture, I have, as the temperature increases, well, so can the activity up to a point, because we know that temperature, increasing temperature, you chemistry people, increasing temperature means you increase the amount of collisions that a molecule can hit each other. If I do that, there's a better chance, the higher the temp, for them to hit and react. Well, that's great up to a point. And then it gets to the point where the enzyme is denatured. And if the enzyme is denatured, well, then it can't do anything. And so then you start to see, as the temperature continues to increase, the activity just drops off, and that there is then no active enzyme because it has been denatured. So temperature, pH does the same thing. pH can most definitely affect an enzyme's capability to be able to work, and denature it just the same. Think about it as almost like burning it, because we know that pH, if we go on the pH scale and become too basic or too acidic, that's going to cause problems as well. You can also have inhibitors that can come into play. Inhibitors can block the reaction site, the active site, or they can change the shape of the active site. And if they do that, then that's also going to impact the enzyme's ability to be able to function. And if we talk about the fact that you can change the concentration of the enzyme, that can help speed up a reaction, as well as substrate concentration. Again, up to a point, because then you get too many and it can't go any faster than it is already working. But if you'll notice that key point there, I like that about the fact saying about optimal range. Because once you have moved out of the optimal range, you're then not going to be able to function. Somehow at that point, it is going to be denatured. So if you look at this little picture real quick, I'm going to give you about 20 seconds. I would like you to look at it, and I'd like you to see if you could fill in those places. All right, all right. Yeah, we're moving on. Okay, so A is representing, yes, the reactant, but we call it a substrate. The substrate is going to go into B, which is the active site. Then we have C, which if you'll notice throughout the whole thing, purple guy doesn't change. He's my enzyme. We are then going to, with the help of the enzyme, this molecule, the substrate, is going to be broken apart. And so what we then get are the products. But you will notice that the substrate changed, but the enzyme itself did not. Now, it is very important to understand that that enzyme has a specific shape for a reason, because it is truly like, probably that your teacher said, a lock and key fit, that it is going to have that exact shape to be able to take care of whatever that substrate is. An inhibitor will mimic the way it looks and can sit in the active site and block it. There's also ways that are called allosteric sites. The allosteric sites are special spots elsewhere on the enzyme. And if a non-competitive inhibitor comes in, it's going to change the shape of that active site as well. It's going to block it from being open. Not that the inhibitor actually goes there. You've got two types of inhibitors. One can actually block the active site, or the other one is going to change the shape of the active site. All right, got to move past that. Now, I'm not going to sit and talk to you about this for any like crazy length of time. I wanted you to have a slide, and it is way too small, in my opinion. I'm getting way too old. There's no way I can read that. But what I wanted you to see is that there are different types of amino acids. And those different types of amino acids are the 20 different ones. They have different things coming off, and you will see on that R group. And I just wanted y'all to see them so that you could see that there are different ones. There are different groups. There are non-polar ones, and there are polar amino acids. And so you have hydrophilic and hydrophobic ones, meaning that when they form their shape, the hydrophobic ones are going to contort and go towards the inside and try to get away from where there would be fluid. Whereas the hydrophilic are going to flip outward, and they're going to try to get closer to the fluid. You've got acidic capabilities. You've got some that have basic capabilities. And then you have some that are just considered neutral. They don't have a charge. So if they have a charge, that for sure is going to tell us that they're in that whole acidic basic area. But anyway, you can go and look at it. I just wanted y'all to see it so that you were aware if you hadn't seen that before. We need to move into nucleic acids so we can get into Unit 2. Yes, we're almost done with Unit 1. Nucleic acids. Very important that you know, anytime we say monomer, we're talking about building blocks. And anytime we're talking about building blocks, there are different components that make that. So in the lipids, it was that to be a monomer, you had to have glycerol and at least one fatty acid. To be a nucleic acid, you must have a phosphate, you must have a sugar. Yes, it's a pentose sugar. To be very honest, that's awesome if you know that. But what I'm excited if you know it's a sugar. What's different between RNA and DNA, of course, is going to be, besides being single-stranded and double-stranded, is going to be because of the fact of the sugar. Is it a sugar that is considered ribose, or is it a sugar that is a deoxyribose, meaning that it has one less oxygen? So the phosphate group is always the same, that never changes. The pentose sugar is going to be the same type of sugar every single time. It's going to be that nitrogenous base. There are 4 different nitrogenous bases for our DNA. And with this, and I know you're going to get more info later when you get to that particular unit later on tonight, but the adenine and thymines always bond together. Y'all know this. Cytosine and guanine always bond together. Y'all know that. It is hydrogen bonding that happens between the 2 different bases. The bases are going to be, you have purines and you have pyrimidines. It is a possibility they could have that. Would they? I could not imagine that. But what you really, and I've seen many times, is the fact of this nitrogenous base. Knowing that this nitrogenous base, is what makes up the middle portion and makes up where the hydrogen bonds occur. There's either 2 or 3 hydrogen bonds between. Cytosine and guanine are going to give us 3 hydrogen bonds. Adenine and thymine are going to have 2 hydrogen bonds. These are very weak because every time DNA gets replicated, they get broken. Every time protein synthesis transcription occurs, they get broken. They get put back together in transcription, but in DNA replication, they actually get ripped apart because you're making strands. When we're looking at what makes that, that's what's called a nucleotide. I've seen it one time on a question, so I'm just going to say it. The side right here, phosphate and sugar. So like on the side right here, this is what's called the nucleoside. I know, because it's the side of it. And then the whole entire thing is called, of course, the nucleotide. And come on. When y'all get to the DNA replication, you're going to end up talking about this, so I'm not going to spend this time. We do know that they run anti-parallel. We do know they are in a 5' to 3' direction. That is huge, and yes, could be talked about. You are expected to know, because when replication or when there is protein synthesis occurring, DNA polymerase and RNA polymerase only work in one direction. They only read in a 3 to 5, and they only add in a 5 to a 3. And so that is going to be something that is important to know, because it is something that goes with 2 different processes that the body does. All right. Last thing is, when we're talking about, actually, I'm going to skip that because we just don't have the time. So I want everybody to stand up. I know I can't see you. I know you can't see me at the moment because I'm sharing my screen. I want you to stand up, reach for the stars. Oh my God. Think about the fact we just got through 1 unit. Got 7 more. Y'all can do it. We can all do it. Take a deep breath. Get yourself a drink of water. None of us probably drink enough water. Can always use water. And we're going to move into Unit 2. Unit 2. With Unit 2, and my watch just told me I had needed to stand up. Perfect. All right, so with Unit 2, we are talking about cell structure and function. With the cell structure and function comes quite a bit of information. I know I didn't do an FRQ with y'all because I had done intro stuff. So once we get through unit 3, I think that's where I got y'all an FRQ. So when we are talking about cell structure and function, College Board tells us that of the whole AP test, we could see 10 to 13% about cell structure and function. No, it doesn't seem like it's a lot. It's the fact that there's 8 units. So chemistry of light, 8 to 11. Now we're 10 to 13. But it's the whole fact that every part of it's important and all of it is close to about the same in the sense of weight. One of the things with cell size, it is super important about that whole surface area to volume ratio. With surface area to volume ratio, we are talking about the fact that the bigger the cell, the more energy it's going to take to make that cell function. It is going to have more waste at that point. It is going to have to move the gases farther. So as our volume increases, the surface area actually decreases. That's crazy, right? But I mean, we've all done stuff in class and we've seen this, but that's why, let's say with our mitochondria, because the mitochondria does our cell respiration, and in the last part of cell respiration, that's where they're in the cristae or in the foldings in the inner membrane, and that's where because of those foldings, they're able to have more electron transport chains. Why is that important? Well, because that means we can make more ATP. So in things like that, the chloroplast, same idea. So if you think about within our stomach, stomach, we've got the rugae, which is a little bit, but let's talk about the intestines, where you've got your villi and the microvilli. Why is that increasing surface area? Why is that important? Because that means that our body can absorb more in a shorter amount of time and in a shorter space. So surface area to volume ratio is super important. You might be asked to calculate it. Do not fret. There is a chart on your formula chart that has it all there for you. But more importantly, it could be that they give you a chart with a few different ones and ask you to give some information about it or ask you to justify based upon some data that they have. But it is important to know that the surface area to volume ratio, that's why the cells stay at a smaller size, why there is those increases in those foldings. I do apologize. I'm at school and I apologize. So y'all get to hear wonderful announcements. I'm so sorry. So anyway, when we are looking at surface area to volume ratio, now let's look at it and the respect of moving to the plasma membrane itself. So we already talked about it being a phospholipid. We already talked about the head being that hydrophilic portion, the polar portion, if you will, the hydrophobic portion there being the nonpolar portion of it, if you will. This gives buoyancy to the cells. This gives shape to cells. The cholesterol that gets added into it helps to give some rigid components so that it maintains a shape. But all of this is important if they ask you about something moving into the cell, if it can come or it can't come, because it's dependent upon if it is a polar or not polar, if it can go through. That also leads to the point where, and I know I talked about that already, about the different types of proteins, that these proteins, you can have proteins that go all the way through. You can have proteins that are embedded, that are peripheral, meaning that they're just slightly embedded, that they then are integral proteins, which are like halfway embedded. But what they can do is they can help in cell communication, which we're going to talk about that. That'll be the last thing that I talk about with y'all. But that's really important as well. is that when you're looking at what is in the cell membrane is going to be there to serve some purpose that the cell needs. So if it is like the two things that shows talked about at the top, a glycoprotein or a glycolipid, these are things that can be, some of those are going to be where these are the receptors, and these are helpful for the body to know that, hey, that's my cell, or hey, this cell needs this particular thing because it has this receptor site on it. So all these things serve a purpose within the cell. Now, oh, water potential. I put this there because I wanted you to know you do not have to know this at all. You do not have to memorize any part of this equation. It will be there. What you do have to know is how to use it. Okay, so let's talk about this first of all. Water potential as a whole is two different things. It is the pressure potential and it is the solute potential. The pressure potential, most of the time, and I say that like 99% of the time, is going to be 0. Why? Because it is in an open container. If it is, oh, I'm so sorry, is in an open container, it is not going to have a pressure potential, which means then if you notice that top equation, that pressure potential just goes away. So the overall water potential is equal to the solute potential. Now, there's a reason I'm going over this, because this really and truly, and I'm going to take some time to go through it, this is something that could definitely show up, okay? And it might be where you have to calculate it. It might not. It might be that they ask you about it, and then you have to make some comments about it in an FRQ, or it's a multiple choice that you have to answer. So let's talk about the equation. The equation for solute potential is based upon 4 different things. The reason why there is a negative sign there, and yes, that negative sign is important because the only thing that has, and it's not positive, it's just 0. The only thing that has a potential of 0 is going to be pure water. As you add a solute into a solution, it becomes more and more negative. We don't have it as being positive. So if it is becoming more and more negative, that's telling us that it is increasing in concentration. So the higher the molarity is for something, the more negative or lower, if you will, on the number scale, will be the solute potential. Ionization constant. This is probably for some of us the hardest part because we don't like chemistry. I do, but we don't like chemistry and we don't remember what we need to do when we see some type of molecule. So if we were told if they just said sucrose and they didn't even give you a formula, folks, they are telling you straight out that it is an ionization constant of 1. Ionization means the ability to split, the ability to become an ion in solution. Non-polar molecules, so like sucrose, they are not going to split. So they, even if, and especially if you saw something like glucose, C6H12O6, oh my Lord, their I is a 24. No. That is not going to be the case. You're going to have an I that's like 1 if it's sucrose, or a nonpolar molecule, or you're going to have it be like 2, 3, 4. You are not going to see like 12 or something like that. Because what happens is if you have a molecule, let's say we had salt. So salt is NaCl. You have a metal and you have a non-metal. And when it goes into solution, it splits into its components, Na plus and Cl minus. So how many ions do I have? I have 2. Well, what if I had Mg, and I apologize, I really thought I had put that in the PowerPoint, and I did not, because I wanted to show you one like, okay, so MgCl2. So envision MgCl2. Well, first of all, there you've got a metal and a non-metal, and it is going to split into Mg, which is only 1, magnesium, there's only 1, and Cl2. Cl2, that's telling me there's 2 chlorines. Well, what that means then is that I would have an ionization constant of 3. You count up how many parts there would be in solution. So if it does not ionize, then your I is a 1. If it ionizes, then you have to count up. And how do you know? If you have a covalent molecule, as in any type of carbohydrate, sugar, or that's usually what they put. It's usually a sugar or a salt is what they have. If it splits up, then you're going to have something with a metal, all right? And so looking at that, I then see molar concentration. Well, in the lab, we might have actually found what the molar concentration is of a solution. Well, if not, they're going to tell you what the molar concentration is. The pressure constant is always that. It's always there. You do not have to worry about it. You just have to use it. You're going to be given temperature, possibly in Kelvin. More than likely, it's going to be in Celsius, and you're going to need to add the 273. But again, you don't have to remember that. It's all there for you. So if we go to, and I don't know how these got out of order, but I want to go to right here. So if I have, go right there. So if I have, it says a student places a potato core into an open beaker containing a 0.55 molar sucrose solution. The temperature of the solution is 25 degrees Celsius. What is the water potential of the sucrose solution? Now, in this particular situation, they did not tell us because they ask us about water potential. So that means that they are asking us about the overall potential. So I need to know the pressure potential. Why I am assuming the pressure potential is 0 is because they didn't tell us anything. Unless you are told that there is a pressure or they give you an indication about a pressure potential, you do assume it is going to be 0. Most of the time, what I've seen is they say it's in an open container. So if that's the case, then we are only going to be solving for the solute potential, because the solute potential will be equal to the overall water potential. So then we plug it in to this equation. Because it was sucrose, it is going to be just the -1. You have your molar concentration, you have your R, and you added the 25, excuse me, to the 273. You are now ready to plug it in. When you plug it in, and yes, of course, you have a calculator, and you would be expected to have a calculator on the test. This is what you would get. Now, what does all that mean, and why was that important? Well, that's what we need to talk about. And I'm sorry I have it out of order. If we were talking about straight osmosis is water moving from a high concentration to a low concentration. What does that mean? If I have a solution that is, let's say, 5 molar solution, and I have a membrane, and it's being separated by a membrane, and on the other side of the membrane, I have a solution that is 10 molar, and only water can pass. I cannot the membrane does not allow for the solute, whatever that solute is, to be able to pass. So that means that on the side that has the 5 molar, there's solutes in it, but there's not nearly as much solutes as there are on the other side. On the other side, where there's 10 molar, there is a lot more solutes. And what does that mean to me? That means that, and I'm going to use all the terms together here now. The side that is 5 molar, and if you want to even like think it in your head, close your eyes and think it in your head. The side that is 5 molar is hypotonic to the side that is 10 molar, that is hypertonic. Why? Hypertonic means that you have more solutes in solution to the compared side that has less solutes. So which way would because that membrane is only going to let water move, the water is not going to move from the 10 to the 5. Ooh, heavens no. The water is going to move from where there is a high concentration of water, not a high concentration of solute. High concentration of water, which would be on the 5 molar side, to a low concentration of water, which is the 10 molar, because it has more solutes. So if I'm talking about water potential now, water potential, if you look at that bullet, or third bullet, it says you can also say that water moves by osmosis from areas of high water potential to low water potential. So if we're thinking about that, and we think about, again, my scenario with 5 molar and 10 molar, the side that is 10 molar, if we were looking at like a water potential value, it's going to be much more negative. It would be, let's say, like a negative 23 bars, whereas the 5 molar side is only going to be like a negative 8 bars. So the water is going to move to the area where there is a lower water potential. So high water potential means that it does not have as much solute as the side where the low water. So it is going to move from the 5 to the 10. All right, so that's one of those things that, and maybe you need to look at it again, but that's one of those things that, yeah, it's come up, but it is something that they could, in a multiple choice question, real quick ask. If we look at this picture, we could say, okay, so if we notice that the middle picture is the hypotonic solution. So it's sitting in a hypotonic solution, and water is going to flow into, and it is going to, because think about like that's a red blood cell, and that red blood cell is just sitting in water. Well, the red blood cell has some solutes in it, but though if it's just sitting in water, that water is going to mass move into that cell until it explodes. Now, on the picture on the left, the hypertonic, that means that now the red blood cell, yeah, it's got salt, but it's sitting in a big old saltwater container and high concentration of salt water. So water is going to move out of the red blood cell into the environment. Even though, yeah, is that going to destroy the cell? Yes, but it is a passive movement of high to low. So it's always going to move from where the water is a higher concentration to where the water is a lower concentration. Unless they talk about, some other solute moving, but it's normally we're talking about water. Now, on the last picture, you'll notice it says no osmotic flow. Well, that doesn't mean like, oh, everybody stop, nobody move. No, the molecules are still moving. It's that there is no net movement in any direction. Because if it is in an isotonic situation, The movement in and out is equal. So there isn't one that is having more or less at that particular time. Can you believe that we have gone through Unit 2? Unit 2, really the big thing in there, yes, you could have different things about the organelles. We're not hitting everything tonight. Please do understand that. When Carrie and I were talking, we wanted to pick the things that were most difficult. And we wanted to pick the things that show up on the test that students seem to struggle with. So that is why we're not hitting on everything. The water potential seems to be one of the harder things. All right, so let me get to the whole picture there. When we are talking, oh, and on this, we are now in cellular energetics, and whoa, doggies, y'all, this is a big unit. We've got photosynthesis, we've got cell respiration, and we're going to come back and talk a little bit more about enzymes. Not much, because I did mention them back there, but they're technically in this particular unit, it's photosynthesis, cell respiration, and enzyme. Just so y'all know, they say this is 12 to 16% on the AP test. So it's got a little bit more chunk to it, but it's also massively huge, and there's a lot going on here. Will they talk about this on the AP test? Yeah, I think so. I always hope they do, because I love both these units, but my thought is that it usually ends up being more the multiple choice than it does the FRQs. But I do have an FRQ for y'all that I am going to show y'all, and it is about cell respiration. So photosynthesis, we know that in photosynthesis, oh, look how pretty that equation is. Oh, that's so nice. It makes it look like it's just one little fun step. Those of us who remember, definitely not one step. Those of us in chemistry know how deceiving that can be. What is going on is a full, what they call redox reaction, meaning that there is the reduction of molecules and there is the oxidation of molecules. Now, it is very, very important, because you could, and they could put it in a sentence and they expect you to know. If they say reduced, that means it gains electrons. If it is oxidized, it loses electrons. They expect you to know that. So in the equation for photosynthesis, as well, the reverse then for cell respiration, what we see is here, CO2 being a gas, is going to get fixed in the plant. And when it gets fixed, it is going to turn into a sugar. It doesn't actually turn into C6H12O6. It turns into G3P. 2 G3Ps make a sugar, though. But the only way that CO2 can get fixed is that the plant has to first go through the light reaction, where they are going to take water, and yes, it does become oxygen when it's split. But the light reaction, the whole point of the light reaction, is honestly not the oxygen. We think it is. But what it really is important is to get the energy that is needed, meaning ATP, and the electrons, the NADPH. It's NADP, and it is going to get reduced to NADPH, and it is going to carry electrons. And that's what's going to help actually make then, in the second part, the Calvin cycle, or the light independent reaction. It is going to make then the sugar that the plant needs. Now, it is amazing to me that I get so tired of plants sometimes. We don't teach plants like we used to, but plants can't get up and move, but they have one of the neatest things, and that is that they have the ability to do photosynthesis, because the ability to sit there and soak up the sun and make food, that's crazy. I love it. Love it, love it, love it. Wish we could do it. So protists, we don't really talk about protists, but we do talk about bacteria. We do know that bacteria there are some bacteria that have photosynthetic capabilities. They are cyanobacteria. They do have the ability with inside to go through to do some photosynthesis. It's really kind of crazy because they are not like a normal eukaryotic cell that has the full chloroplast, but they are still able to do some photosynthesis. And then, of course, we also have, like I said, the protists. We just don't really talk about them, unfortunately, anymore. But they, of course, they are eukaryotes, and they do have full chloroplasts in them. But if we are talking about the process again, there's two big things. You've got to do the energy capturing and getting stage, and then you're going to, I should say, harvesting, and then you're going to go and use it. So the light dependent, and both of them happen in the chloroplast, but within the mesophyll region of a plant cell. So it's only one little special place. And what's crazy is the chloroplast itself is not green. It's in the thylakoid membrane where you find the chlorophyll A and B. That's crazy. But that is where in the thylakoid, in the membrane of the thylakoid, this is where we are going to see the part where the sugar ends up getting, well, in the Calvin cycle, right outside the thylakoid membrane. Now, the first part, and I have another, I wanted to show you these pictures. I do like these pictures because this is showing you that mesophyll region. And it's showing you not only in this mesophyll region, it's showing you then how that chloroplast and what that chloroplast looks like, and how it has a double membrane. Chloroplasts have their own set of DNA as well. But you see these stacks of thylakoids, and they look like these discs, and they're interconnected. There's just fluid inside, but this is where the protons do move inside for a little bit, and the protons then move back out as they go through an electron transport chain. But this picture will, and I'm going to come back to that one a second. I like this picture because this picture lets us kind of see it zooming in and a cross section of a thylakoid. So this is the actual cross section of a thylakoid here. Well, not actual, it's a drawing of the thylakoid. And you can see that you have a phospholipid bilayer, a full phospholipid bilayer. You have my favorite molecule, ATP synthase. You have the electron transport chain going on right here. And then you have photosystem 2 and photosystem 1. These are actually where the light harvesting pigments are. That's where the green actually is. It's nowhere else. But isn't it crazy that I think it's so amazing? That little place and the plant looks green. Pretty interesting. All right, so I'm going to go back over here for a second. So when I look at this, we are going to go through 2 photosystems to be able to get to, and I should be, as we know, this should be review right now. This should not be, I'm teaching this to you. So as I'm saying these things, hopefully you're thinking about, oh, yes, I remember. Okay, so we have P680 and P700. Remember, those are the photosystems name. They're the reaction centers. In the first reaction center, when photons, which the energy associated with the wavelengths of light, when they hit those light harvesting units, they then ignite, if you will, excite 2 electrons to move up to an excited state. As these electrons move up to an excited state, we hopefully know that electrons When they jump up to an excited state, they will fall. And when they fall, if nothing catches them, they will release their energy, and they will release that energy as heat. And so that could be a huge problem, but that's actually perfect because the electron transport chain, which is being represented right here by just arrows, that electron transport chain is going to help power, those electrons are going to help power the chain to help make ATP. Super important. We're going to make ATP in between P680 and P700. So then those electrons, what's so cool is they finish the electron transport chain and they now have lost their energy and they're now at ground state. So they fall into the P700 reaction site. Why? Because photons were hitting them too. And when photons hit the reaction site, electrons jump up to an excited state. We have a huge problem if electrons leave and they are not replaced. Why? Because then that becomes unstable. So back here at P680, photons hit, like it shows, light energy hits the P680. When it hits P680, y'all, and they jump up to an excited state, the photosystem, the reaction center of the photosystem is now unstable. This is where the plant does what's most important to us is that it splits water. And in the splitting of water, this is what's so ingenious to me. When it splits the water, the oxygen will end up leaving out as the oxygen out of the stomata becomes the oxygen that we breathe. Those protons make a solution acidic, not good. But in this case, those protons are going to help what are going to move through the electron transport chain. And because, of course, and my students know, my students know I put it all up here for us, so I don't want us going through it like that. My most favorite molecule in all of the world is ATP synthase. ATP synthase is so freaking cool. When the protons, the reason those protons are so important from the splitting of water is because those protons are going to move through the electron transport chain, but then they are going to move through ATP synthase. And when they move through ATP synthase, they are going to cause ATP synthase to spin, and that's going to make ATP. Oh my gosh, it's so cool. But back to the light energy, when those electrons move to an excited state, when water is split, the electrons then fill the reaction centers whole. Then you get to, after the electron transport chain, you have your next one. Well, you have this molecule, and it's NADP. NADP is an electron shuttler. He can pick up electrons, and when he picks up electrons, he is able then to carry those electrons and actually protons. And when he carries those electrons and protons, he's actually going to help carry them to the Calvin cycle. Now, what's so important about that is the fact that when NADP carries electrons, they are still considered at a high energy state, which is important because they're going to then help in the making of the molecules, of the sugar molecules in the next portion. Now, in all of this, when we go through what this is called the light-dependent reaction, when we go through this part right here, this will make equal amounts. And we don't, unlike in cell respiration, I always kind of go through the numbers just so I can talk about them. But when we are talking about photosynthesis, you have to understand that it makes equal amounts of ATP and NADPH. That's super, super important, okay? Because now we're getting ready to go to the light independent or Calvin cycle, and we're going to use ATP and NADPH that was made here to help make a sugar. But this process does not use equal amounts of NADPH and ATP. And that's where the problem lies, because then what happens is, and let's go to, so right here, we can see that the CO2 comes in, and of course the CO2 comes in from the environment through the stomata, and this is what's called carbon fixation. There is an enzyme that is called Ribisco, who is going to help bind with a molecule called RubyP, and they are going to go through, as I tell my students, magic, because it is going to go through many steps that we are not required to know. And it is going to fix the CO2. So with the help of ATP and NADPH, hopefully you can see in this picture that you are using equal amounts of ATP and NADPH. That's awesome, because what that makes is 1G3P. What it then does is technically right here at this point where that 1G3P comes out, there were technically 6 G3Ps. But for 6 G3Ps, that would only give us 3 glucose molecules, and I would not have a molecule that gets to get regenerated, which is called RubyP. RubyP is the starting and the ending of the Calvin cycle. Just like oxaloacetate is the beginning and the end of the Krebs cycle, and that you have this regenerated molecule. If the plant used all 6 of those G3Ps, it would have to go make more RubyP. It obviously takes a lot more energy to make RubyP than it does G3P, because it then takes those 5 G3Ps and regenerates them with the help of more ATP. And it becomes RubyP again. So what's really important to know, what we put in, which were 3 carbons, we got out 3 carbons. What you put in is what you get out. Now, the unfortunate reality is we used an equal amount of ATP and NADPH for the first part. But to get it back to the regenerated portion, we then had to use more ATP. And now we've got a problem. Because what's going to end up happening is there's going to be a deficiency. And the deficiency is going to mean that there is not enough, because the reason why this is called a light independent reaction is due to the fact that this particular part, there is no light energy that's hitting this. All that this is dependent upon, this entire cycle, is there enough ATP and is there enough NADPH? And if there's enough of those fellas, heck, we're good to go. We can keep going, and it goes on into the nighttime. But at some point, it's going to run out. And when it runs out, now it doesn't matter if it's daylight or nighttime, it cannot do this cycle anymore. So now we need to go back, all these things, and we need to look at this right here. So this is what normally happens. P680 is also called photosystem 2. P700 is called photosystem 1. There's a reason for that, not to confuse you, but because scientists found P700 first. What that means is that the plant obviously becomes deficient on ATP a lot, because what happens is there is an inhibitor that comes and blocks the reaction center P680. and blocks the ability for electrons to go to NADP. If these are blocked, and the photons are hitting P680, but there is an inhibitor that has stopped the reaction from occurring. What happens is that this fellow right here, this reaction center, P700, it is going to get the light energy, and that light energy is then going to ignite it, and those are going to be high energy electrons. But wait a second, they normally go this way. There's now been a block that's put on that. If there's a block right here, where's it going to go? It's going to go backwards. And it's going to go backwards through what is being shown here is just little arrows, but it is going to go back through here, and it is going to go through, because what is here? An electron transport chain with my favorite molecule, ATP synthase. And so ATP synthase is going to make ATP. So what happens is when there becomes a deficiency on ATP, because over here, and this part right here, this only can happen, one, light. This also cannot happen, the part that I was just talking about is not going to happen when the levels are high. When the levels are high of ATP and NADPH, then you're going to have this whole cycle and then the whole Calvin cycle. But when the levels of ATP fall too low, and if this happens at night, well, then it all stops until morning time. Because what has to happen here is there has to be photons. And when the photons hit right here in P700, they jump up to an excited state. They then go through the electron transport chain, and this is what's so cool. They fill the hole that they created, and they go right back into the reaction center. Then the photons hit them again, and they jump up, and they go through the electron transport chain, and they make more ATP. And they continue to do that until the levels of ATP have gotten back up. Then the inhibitor is removed from right over here that's blocking NADP, and from over here that's blocking P680. So this is why they call this P700 photosystem 1, because scientists found that one first in the discovery, but the problem in that is it's what they then found is it's not the first one, because in the big scheme of it, P680 is first. But they found this one because deficiency happens quite a bit. Okay. And I hope y'all are giving Jed some questions so that he's got something to do. Hopefully everything's okay on that end. I feel weird. I hate talking into a screen and not getting to see people. I hate that. All right, so let's move from good old photosynthesis. I like photosynthesis, but to be honest, I love cell respiration. So this is important. And I guess I'll say that going backwards in a sense for photosynthesis, it is important to know where they occur. So in photosynthesis, the light-dependent reaction, so the first part where you're making ATP and NADPH, is going to happen in the thylakoid membrane. It's going to go across the thylakoid membrane into the thylakoid, meaning the proton, it being the protons, excuse me. And then the protons come out of ATP synthase into what's called the stroma. In a eukaryotic cell, a non-plant cell, we call it the cytoplasm. Well, in a plant cell, inside the chloroplast, we call it the stroma. So the stroma is the area inside the chloroplast, not inside the normal portion of the cell, but because all of it happens encased in that chloroplast. So the stroma is where then, because the protons came out there, that is going to be where the Calvin cycle or the light independent reaction occurs. It's going to happen inside the stroma, outside of the thylakoid. Okay, so now let's talk about in cell respiration. Cell respiration takes place in two different, well, technically kind of three. Glycolysis happens in the cytoplasm, so outside of the mitochondria. Then, as we move into heading into the Krebs cycle, we're going to be moving into the matrix of the mitochondria. Remembering there is kind of a little intermediate step that is going to happen before we actually enter into the citric acid cycle. The citric acid cycle, or the Krebs, takes place in the matrix, so the very, very inner cavernous portion of the mitochondria. And then the last part is going to take place in the inner membrane. And the inner membrane is where we're going to have, and now I know it says electron transport chain, but I want to use the technical word there. The electron transport chain is the place where it happens. The actual process is called oxidative phosphorylation. Oxidative means loss of electrons. Phosphorylation, making of ATP. Through the loss of electrons, who's losing those electrons? Woo, doggies, glycolysis. Glucose is losing them. And through glycolysis and the Krebs cycle, glucose is going to be completely stripped clean of its carbons and its electrons. And all of that then is going to be transferred over so that we can make ATP. Okay, so in this picture, you can see what we have here showing us the different places. And that is also important to see how there is this difference here. So glycolysis, as I also tell my students, it's magic. No, not really. There's like 10 steps it takes for glycolysis to go through to completion. But we do not have to know those 10 steps anymore. Thank goodness. When I first started teaching it, we did. But what we do need to know is that we are going to have to use a little energy to be able to get energy. And then we are going to be able to break glucose in half, is really what kind of happens. Literally kind of just snaps in half. Not really, because there's all those steps. Then we make a tiny bit of energy in glycolysis. Then we're going to move into the then we're going to move into the citric acid cycle. But before we can get to the citric acid cycle, pyruvate is not a reactive enough molecule. It's going to get a coenzyme attached to it. And as this coenzyme gets attached to it, I always call it the intermediate step, but a lot of places call it the finishing of glycolysis. I mean, glycolysis is literally just the pyruvate. But then you move where you have to, because you cannot do the acetyl-CoA portion unless you have oxygen. And you're moving into mitochondria at that point. Then you go through the citric acid cycle, and then you're going to be able to finally get to the electron transport chain. Now, you'll notice at the bottom of the picture, it's a substrate level phosphorylation and oxidative phosphorylation. Now, that's big, and that is the difference. Substrate level phosphorylation, it means basically the breaking of a molecule gets you some energy. Oxidative phosphorylation means through the loss of electrons, you're going to make energy. And so this is more of like an indirect pathway. But this indirect pathway is going to be much better in the long run. So now let's look at what happens. Is it not working? There we go. So again, we're in the cytoplasm for the first part. And so you'll notice in this picture, and this was actually a picture from the AP test one year. What we have right here at the top, glucose is being shown as a 6-carbon molecule, like it is. It's 6 little circles here. And what we have is that we had to invest some energy. So making sure that y'all see that ADP. So we have, it doesn't actually show that. I apologize. It does not show that we had to put it in, but you did. You actually had to put in 2 ATP. As a result, you actually get 4 ATP, but you only get to net 2, which is why the picture shows this. So we made 2 ATP. We made 2, now NAD is like NADP. It is an electron shuttler. It is a reusable molecule over and over again, and it is going to shuttle electrons over to the electron transport chain. When it does this, it picks up electrons and hydrogen ions. Hydrogen ions and protons, of course, are the same. When we're done, you will notice that we still have 3 carbons and 3 carbons because you'll notice it says 2 pyruvates. So we still have all 6 carbons of glucose, but we have gained a little energy and we have gotten some electrons. Now, the whole first glycolysis, the intermediate, and the cycle, all 3 of those, we will see that every carbon of glucose is going to be taken away. Every single carbon molecule is going to leave, and it's going to leave out as CO2. We're going to see that we're going to gain electrons as well. Those electrons are going to be what powers to make us that massive amount of ATP. So you'll notice that was a very, very small amount of ATP that we actually made right here. For sure, not enough to keep everything moving. Now, as we go from pyruvate to acetyl-CoA, this is happening as we're moving into the mitochondria. There will be a coenzyme A that gets attached. There, you'll also notice right here, we get from each pyruvate, we are going to get a carbon that's going to come off, and it's going to be a CO2 molecule. Okay. Then we're also going to get some electrons and some protons here. So, and those protons, they're coming from glucose. So we are getting the carbons, the oxygens, and the hydrogens are all getting stripped away, and they are then becoming or going to somewhere else. The CO2s leave out completely, or waste, they leave out. So the oxygen that is used for cell respiration does not come from glucose. The oxygen that is used for cell respiration is found from the oxygen that we breathe. So when we're done with glycolysis and the pyruvate portion of, I should say, the changing at the intermediate step that I call it, we are now going to have acetyl coenzyme A. This acetyl coenzyme A is now ready to go into the Krebs cycle. And where is that going to happen? Inside the mitochondria, inside the most inner portion, and that is going to be the mitochondrial matrix. Okay, so now let's go to, we do not have enough oxygen. If we do not have enough oxygen, if there's not oxygen, there is not going to be then moving into the mitochondria where we do that little intermediate step. Instead, what is going to happen, you'll notice, okay, so let's start with, we'll do the lactic acid over here first. We've got glucose, it goes through glycolysis, yay, and we get our pyruvate, yay. In the process, we made 2 ATP and we made 2 NADH. Yay, wonderful. Normally, the pyruvate is now going to move into the mitochondria. If there is not oxygen, instead what's going to happen is that these NADHs, they are going to come over here and they're going to work with pyruvate and they're going to rearrange themselves into lactate. And now we've used up the NADH that had electrons and protons, and we've lost pyruvate. And now it's done. You gained 2 ATPs. Who's thinking ATPs, people? You did not do enough to be able to maintain. And what's going to happen is that lactic acid starts to build up in the cells. Now, and the reason they say fermentation, y'all, it literally is like the cells are getting drunk. Because that is something now that has to be broken down by the body. The body has to break that down. Now, over here, let's look on the other side. Alcohol fermentation, it's slightly different, still very, very similar, but you'll notice that the glucose goes and through glycolysis, yay, we made our pyruvate, yay, we made our ATP, yay, we made our NADH. Then what happens is that it is now going to actually, because pyruvate is a 3-carbon molecule, it is actually going to break off one of the carbons. And then it is going to add it to the, it is going to, the NADH is going to then give the electrons and the protons to this 2-carbon molecule, which we're calling acetaldehyde. You do not need to know the name, do not worry. And it then turns into ethanol. Ethanol and lactate is a slightly different process, but what's important to understand is this is what then happens to plant cells or animal cells in the absence of oxygen. This is not long haul enough to sustain the cells, okay? And that's why we call it fermentation. So now we move into the electron transport chain. The electron transport chain. Oh, I love that. The electron transport chain. We are in the inner portion because we finished up the, and I did not, I'm so sorry. I flew over the Krebs cycle. Acetyl-CoAs, assuming that we do have oxygen, they have, the pyruvates were turned into acetyl-CoA. Now what's going to happen is they are going to enter into a cycle that uses a regenerative molecule just like we had in photosynthesis. So there is a molecule called oxaloacetate. No, it's not important that you know it, but what's important to know is that acetyl-CoA and this regenerated molecule that gets used over and over again, which that's oxaloacetate, they will react together and they will go through a series of crazy steps that we do not have to know. So it's magic. There is going to be CO2s that are going to come off. There were 2 carbons that entered this cycle, which was acetyl-CoA. Those 2 carbons are going to come off. The coenzyme A is going to come off, gets reused. The 2 carbons from the original glucose are going to be broken off as CO2. You have now, all of the 6 glucoses are now going to be gone, or 6 carbons of glucose are all going to be gone. Then you're going to have the NADHs, which means you had electrons and protons that are now going to be carried by a shuttle. I call it a shuttle bus. It just makes more sense in my head. And so the NAD is going to pick up the protons and the electrons, and it is then going to move them over to the electron transport chain. Then you're going to have, there is a cousin to NAD, if you will, and it is called FAD. FAD is a shuttle bus as well. It picks up electrons and protons and shuttles them. It's not as efficient. Every one NADH converting wise converts into about 3 ATP, whereas one FADH2 is going to probably convert into 2 ATP. So back to the whole idea. We are in the matrix of the mitochondria. Every one of the 6 original carbons of glucose are now gone. We haven't made like hardly any ATP. We made a little bit here, GTP. It's basically the same as ATP, and we made some in glycolysis. We want to make like 38. Oh my goodness! Yeah, we need to step that up. But that's the problem. Substrate level phosphorylation does not give us a lot of ATP. You break a molecule, you get a little energy. You don't get much. What ends up happening, though, is we have collected all of this NADH and FADH2. So if you notice right up here, we got some. Right over here, we got some. And now we're ready to take them to the end place. And that's my favorite, literally one of my favorites. So we are now in the mitochondria inner portion. Matrix is where all the NADHs and FADH2s are located. They are sitting in the matrix of the mitochondria, ready to drop off electrons. Who are they going to drop them off to? Electron transport chain. Where is the electron transport chain? In the inner membrane of the mitochondria. So they then, and you'll notice in this picture right here, on the left-hand side, you see NADH is going to get oxidized. Oxidized. Yeah, that means the loss of electrons. It is going to lose its electrons. The chain is going to technically gain the electrons, if you will, for a little bit. And the electrons are going to move through this electron transport chain. Now, here's the deal. They are high energy electrons. When they have high energy, they are going to be able to give these, what you'll notice it says like complex 1, complex 2, 3, and 4. Those proton pumps, they're pumps, but they're normally closed. They pump protons only if they're open. High energy electrons are going to give them just enough energy to open for a little, and then they shut back. But they open long enough to allow protons to pass across. Let's back that up. Wait a second. Protons, where did they come from? They came from NADH. They were carrying the protons too. As these protons are building up over here, y'all, we are building up acidity on the inner portion in the matrix. That's not good. We have a high concentration of protons. Now those pumps open and those protons are like, I'm moving, because they're going to spread out. And this is why it's called chemiosmosis. This is the portion of it. The protons are moving across from a high to a low concentration. So now they've moved across. Well, that doesn't seem very smart. We just moved acidity from one side to the other. So the protons move across. Now they are in the inner membrane space. So they're in between the outer portion of the mitochondria and the inner membrane. We've done nothing, but actually we've done quite a bit. So now what's going to happen is, and if you'll notice on the other side right here, I've got this picture of this mitochondria, and I will tell you that is a sad picture of mitochondria, of a ATP synthase, because ATP synthase is my most favorite molecule ever, and it is so cool. It looks like a windmill, because what ends up happening is these protons are now going to move across. Why? Because this is a one-way mover. The ATP synthase, when those protons were in the matrix, they couldn't go into ATP synthase. ATP synthase has, technically, it's like blades, and these blades move just like a windmill. As these protons move through the channel here, they're going to cause ATP synthase to start to spin. As ATP synthase starts, this is so cool. If you don't think this is cool, something's wrong with it. They start to spin. And as ATP synthase starts to spin, it is then going to the blades are able to take ADP and inorganic phosphate, and they throw them together with enough energy. They throw them together with enough force that the electron clouds overlap and bond together without causing energy, without using energy. So this is why it's oxidative phosphorylation, because through the loss of the electrons from NADH, it powers this whole process to make ATP. And here's the best. Well, no, ATP synthase is the best part. But as the electrons moved through the electron transport chain, through those proton pumps, they finish and they have now released their energy. So they're now at ground state. They're going to go, and this is why they move. They don't just like, I'm going to move through here to here. No, they are literally pulled to oxygen, who is at the end of the electron transport chain. Oxygen is the second highest electronegative atom that we have. Fluorine is the highest. Oxygen is next. Oxygen wants electrons. Why? It helps to fill up the outer shell. Now, it wants electrons. Those electrons are being pulled to it as those electrons, and y'all, we cannot just have electrons floating around somewhere unattended. Not a good idea. Like small children, you can't leave them unattended. So the oxygen and the electrons are going to come together, and here's the best part of it. Well, those protons, we moved them from the matrix to the inner membrane space through ATP synthase, the best part. But now they're just there. And that would cause an acidity issue, but it doesn't, because the oxygen and the electrons are going to bond with the hydrogen to make water. And in all of this, yeah, it's technically considered a waste product, but our body needs so much water, it's really usually not a problem. And now, I mean, and since if it's never a problem, our body can just excrete it if it's extra. But now it has taken care of the electrons, it's taking care of the protons, the carbons from glucose left out as CO2, and in the end, we made ATP. We made a massive amount of ATP. So through a technically what's considered an indirect method, we were able to make a whole bunch of ATP. How can you not say that's cool? It's so cool. It is. It's amazing. All right, so I see that I have 30 minutes, and I am, as much as I want to do this FRQ, I know y'all have the PowerPoint, and I have that right there for y'all. I need to move into cell communication. But before we do that, let's stretch ourselves. We are not meant to sit this long, people. Too bad I don't have like a cart that I could just like roll my computer around my room while I'm talking to y'all. Like, steps in, and then I could not be so stiff. All right. Take a drink of water. I'm sure my students who are here listening are rolling their eyes at me, but that's okay. Love y'all too. Okay, so we are now into Unit 4. And Unit 4 is cell communication and cell cycle. With cell communication and cell cycle, it's about 10 to 15%. In my opinion, that's kind of a big range. It will show up on the test, y'all. There's no question. It will show up on the test. To what degree? But it will. And my guess is about the signal transduction pathway. Is it going to talk to us about probably the short term or short distance? No, probably not. It's probably going to be the long distance communication. But regardless of it, there are 3 different pictures that are here. And these 3 different pictures that are here are talking to you about in the respect of bacteria, quorum sensing, Bonnie Bassler and quorum sensing. Amazing. Talking about how bacteria talk to each other. The picture on the left-hand side is a neuron, and talking to you about how a neuron, the signals, and this is very close distance signaling from one neuron to the next neuron, how the chemical signal moves to be able to continue the pathway. And then here, looking at bottom one, talking about, excuse me, in the respect of doing a negative feedback system. A negative feedback system, super important. Most of the feedback systems in our body are negative. We do have some positive feedbacks, childbirth, blood clotting, but most of the time, like this figure, what you see is that if our blood sugar goes up too high or goes down too low, we are going to have the release of hormones, and those are then going to initiate different things to occur from the liver. So the liver is either going to take in the glucose and store it as glycogen, or it is going to break down that glycogen and release it out into the blood, so the blood glucose level goes back up. In this, you have technically they're like, I mean, they're signals is what they are, but we call the signals ligand. And I say ligand, I don't say ligand. Just sounds weird to me. Actually, they both sound weird, but I like ligand better. I think it's because I'm from Texas. So ligand is going to be the signal that is going to be sent, and the receptor is going to be then what picks it up. And the receptor is most of the time, not all of the time, most of the time, the receptors are on the outside of the cell. Sometimes the receptors, as in the case of steroid hormones, they are on the inside of the cell. And that means they actually have to go into the cell first. And that's because they're lipid-based, mainly. And they are going to go in first before they start their pathway. But let's look at, in the respect of here, there are different ways that cells can get a signal. They can get a signal from itself, which would be an autocrine type of way. They can signal cells that are quite close to each other. Sometimes they're actually attached to each other like that. And that would be then across a gap junction. But if they're a little distance away, that would be like a paracrine signal. Or if they're really far away, that means it needs to go through the bloodstream. And that's what we would consider the endocrine type of cell communication. But regardless of it, with any of this type of signaling, that it is important to know that the signal is moving not through its own channel. Okay, like in the case of the gap, yes, it's like from the channel protein. But what I meant by that is it doesn't have its own highway system. It's going to be flowing either very close in the interstitial fluid, or it is going to be traveling through the bloodstream. Okay, so next thing. Hello. I don't know what's going on there. When we're talking about like in plants, plants have the plasmodesmatas, and these are going to be channels because they are, yes, they do have xylem and phloem, but they don't have a full highway system. And so, well, like we have our circulatory system. These are channels that are actually between each one of the cells. And so that way things can get from one place to another. In the immune system, we do have direct cell-to-cell contact, and the immune system is one that does that. The immune system does a very good job of that. And this is how, when they are talking to and trying to figure out type of pathogen that's in the body, those receptors are going to be working on that. Okay. Now, this shows you a little bit better in the respect of kind of like how in the nervous system, how it would look with a neuron. You see right here, this is the ligand. So this is the signal. What we would actually call this in the nervous system is this would be a neurotransmitter. This neurotransmitter is actually going to have to get released out of the pre-neuron, and it is going to have to be released into this very, very small region. But it is there. You'll notice they're not connected. And then what's going to happen is there are receptors on the next neuron, the host neuron, and that is then going to help for the signal to continue on. So in looking at this, you have receptors, you have the ligand, and they are just going to look a little different. Now, in signal transduction pathways, so if we're talking about really like the most of our long distance signaling that we're looking at in the endocrine system, there's some type of reception that's going to have to be done. Then there's going to have to be some type of transduction that's going to have to occur. And then, instead of doing a slide for each one of these, because that in itself could be slightly overwhelming, I put it into a chart for y'all. In all of this, though, it leads to a response. The response could be it's time for the cell to grow and divide. It could be that it is telling the cell that this particular cell needs to make a protein. And whatever that protein's job is, it is needed. And so cells do not do things on their own accord. If they're doing something on their own accord, meaning that there has been probably some kind of mutation that has led to them doing things on their own accord, because they should 100% be following the signals that they receive to be able to start and stop things. So if you'll notice on this next one, I went ahead and I gave you the different types. So you'll notice I'm going to do this last one at the bottom. This is what I was saying about how most of the signals that are received by a cell are received by a cell on the outside. There's a receptor on the outside at the cell membrane. The one that is different is this is the intracellular receptor. And this is going to be, it's actually going to start in the cytoplasm. And it has to be a ligand that is non-polar. So we're looking at our steroid-based hormones. They're, of course, not all non-polar. And what their pathway is to that in this particular, that the transcription factors, which means that it's going to inside the cell to express a gene. So it is not, the cell's not needing to divide or anything like that, but it is needing to, something inside that particular cell is needing to be produced. And so that is going to happen through that gene expression. Now, the ligand-gated ion channels, I don't want to say it's the most common type, but it's the one you see more often. When I've seen different things on previous AP tests, it was more of those, but doesn't mean the other ones could not be talked about. They're going to be, the signal is going to come to the cell membrane, and it is going to have to travel across the cell membrane, but it's going to need a receptor to be able to start all of that. And I gave you an example being stimulation of neurons. The G protein, coupled receptor, this is going to have, then this is got where it gets to the inside, then it goes to secondary messengers that are inside that leads to usually a few steps that will, and like an example of this one, so adrenaline was the example that then leads to the contraction of smooth muscles. Then we have okay. Then we have the receptor tyrosine kinases, and this is going to lead to where you have several steps. This is but this is the one you see a lot in like over here in that picture I had. That is kind of like this whole transduction pathway is a lot of times what when they're talking about the phosphorylation cascade. And so there's going to need to be several steps. There's going to usually have to be activation of different molecules. There's going to be energy a lot of times that's associated with needing to be used. And then that's going to lead to then, in the case I gave you, cell division. All right, so now when talking about these are just common responses for sure. Gene expression, oh my gosh, that's how they get activated. Enzyme activation, and then apoptosis. So programmed cell death is now when we have cells, and there are, I shouldn't say cells, when we have specific genes that get mutated, they then won't follow this programmed cell death. And this could be a serious problem as well, because it can lead to other problems. So let's talk about my last part. And then, oh my gosh, I might be done early. I don't know. That's kind of crazy. I'll find something. I won't let you have my time. Just kidding. So we have in the when we're talking about cell cycle. So if you will notice that you have this large orange portion that is interphase. Interphase is made up of G1, S, and G2. G1 and G2 both have growth. Final preparatory steps are going to happen in G2 that would not in G1. You will notice that G1 is a lot longer than G2 because there's going to be in the respect of organelles and fluid and the cell membrane. Because y'all think about it. If you've got a certain size with, let's say, 800,000 phospholipids, yeah, it's a lot more than that, you're going to need to double that before you are ready to get into mitosis. Now, mitosis is specifically just cell division. Now, within that mitosis, there's also going to be the cytokinesis. Sometimes I've seen it where cytokinesis is actually like its own little bitty strip because technically cytokinesis is the cell splitting. Telophase is which is the last step of regular mitosis. Telophase and cytokinesis kind of start happening together. And that's where telophase, the nucleus is getting back together. It is incorporating all of the chromosomes and all of that. Whereas cytokinesis is literally the splitting of all the fluid and the organelles and doing either a cleavage through if it's an animal cell or making of a cell plate if it is a plant cell. But in this G1, S, and G2, you'll notice that the synthesis of the DNA takes a long time. And especially in our body, a eukaryotic cell, it takes longer than it does in a prokaryotic cell. And that's because of all the proofreading that happens within the synthesis portion of a eukaryotic cell. Now, there are cells that move into a G0 stage. That means they're exiting the cell cycle. They do not replicate anymore. By definition, a neuron, once it has reached maturity, it does not divide. It is, by definition, in G0. So if it is destroyed, by definition, it is then done. Now, we know that there are regenerative capabilities. We know that with gene therapy and things like this and blah, 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 but I'm just saying by straight definition. A muscle cell, by definition, if a muscle cell goes into G0 as well, and it is not making more. When your muscle cells are building and becoming stronger and doing more, it's because of the actin and the myosin, which I don't expect you to know about, just throwing it out there. It's because they're building more of the things that help the muscle contract. And so it's not that the muscle is actually getting torn and rebuilding on it. It is building more of itself. So there are some cells that go into a G0 state, which means they are then not going to be doing. So like in anatomy, we finished not that long ago talking about the fact that when in reproduction for meiosis, that in females, all our eggs go into a state of G0 until they are actually released from the ovary. And so they were made we were in the womb, and then they go into a state of dormancy, and they're in a state of G0 until they are actually released from the ovary, if they're released from the ovary. So when we look at this is just the cycle itself. Well, there's also these checkpoints along the way. And these checkpoints help to ensure that everything that happens when it's supposed to. Everything that if there has been a problem, if there has been a mess up, that it stops. You'll notice that after the growth stage right here, you have your first checkpoint. You don't have a point right here. You have a checkpoint, a G2 checkpoint. But y'all, this one is super important because if there was a mess up with DNA, if things didn't fix themselves, it will go into programmed cell death here. And it should go into apoptosis. Then in mitosis, if it doesn't line up correctly, it is supposed to go into, again, if it doesn't pass that checkpoint, it is supposed to go into apoptosis. So this would be a really good place for them to throw something at you about a disruption and what would happen if that this particular place, and if you have one thing happening versus another thing happening, that they could throw that at y'all. All right, so I did put in, and this I think is my last slide, experimental design. If, and they have, no question, if they ask you about designing an experiment, if they ask you to give parts to an experiment, expect that. Just expect it now. Let's look at this particular question. What you need to know, they do not want you to give them the procedure for an entire experiment. You don't have the time for that. They want if they give you, like in this case, it says design an experiment using artificial flowers to investigate potential negative effects of increasing caffeine concentration in nectars on the number of floral visits by bees. Because this was a whole like bee question. There was a whole bunch of stuff they had to graph at first, but they want you to identify the null hypothesis, what would be a control, and what would be results. And notice what they said here. It could be used to reject the null hypothesis. That's what they're going to want you to do. And now let's ask, what is a null hypothesis? And I know Carrie's going to go into that with y'all. But what is a null hypothesis? Null hypothesis means that there is no difference between what is expected and what happens. And so in the case of this, you'll notice it says using artificial flowers to investigate potential negative effects of increasing caffeine. Well, there would be no effect on the bees by increasing the concentration of the caffeine, that it would not affect the outcome. It would not affect the, and they put in parentheses, the kids did not have to say number of floral visits. But what they wanted to know is that there would be no effect. There would be no difference. And so what would the control be is that you're thinking about, so the control is always, so I like to think about placebos, because like my two of my friends on either side of me at my school were both in the clinical trials for the COVID vaccine. One of them actually got the vaccine. The other one was given a placebo. Well, why is that important? Because they need to see that what different reactions can occur and what shouldn't happen and what does happen. You always have to have the control. So in the case of this, it would be nectar that doesn't have caffeine, because they want to see, is there any negative effects to it? And then predict results. Now, predict results, notice this, that they said used to reject the null hypothesis. Rejecting the null hypothesis means that you are going to say there was something going on, not that there's no difference. So the number of floral bees is different at increasing levels. That it was different from the control, that there was a negative effect to it, that it changed the number of visits at increasing the caffeine, but all of it had to be about the fact that you were rejecting the null hypothesis. So understand that on the test, if they do ask you about experimental design, they're going to give you, like in this case, about the artificial flowers, investigating negative effects. They're going to do that, but then they are going to have you come up with the rest. They weren't concerned about what was the procedure of how you will do that. We can get your ideas of what you're going to do just by those three things right there. And with that, I am now finished. And we've gone through 2.5 hours. I'm so proud of y'all.
Okay, here's my face, everyone. Let's see if we can get this going. All right, I guess I'm super bummed. But anyways, that's okay. I was hoping to use this software tonight where you could all see my face, and then I could also do some drawings for you. So maybe the people who help would be able to help us out. But anyway, so I will just get started doing it the original way, where I just show you my my slides. And so maybe if they help me figure out a different way to do it, we can do that. So anyway, just give me a moment. Make sure my captions are on. English. Okay, here we go. So I'm Mrs. Parnan. If you haven't had me as a streamer before, I apologize that my video and my face is not there. That is what I had practiced today to make sure that you'd be able to see me and also be able to see some other drawings that I was going to be doing, but I can't, for some reason, I guess that's not going to work. So I really apologize for that. It's such a bummer. But anyways, let's just move on. We're going to tackle heredity first. So when we think about heredity and traits being passed down, you think about the very beginning of an organism's life, if they're a sexually reproducing organism, and you have to think, well, that came from two gametes. There was a fertilization event. And we think back to, well, where did those gametes come from? That's where meiosis comes into play. And the big things that you need to be able to do is compare and contrast mitosis with meiosis. They sound very similar, but alas, we learned about mitosis during the cell cycle because we're creating identical cells, right? They get signaled to divide, just like Anne was telling you. Those signals are super important. So if I get a paper cut, for example, which I do all the time because I'm a teacher and I'm constantly shuffling papers, if I get a paper cut, those skin cells are going to be signaled to make identical skin cells for me. There's one division. We're going from diploid to diploid, so we're not changing ploidy at all. And remember, ploidy just has to do with the sets of chromosomes. So if it's 2N, you have 2 sets. So for organisms like us, right, our 2N is 46 chromosomes because 23 came from mom, 23 came from dad. Now with meiosis, we're creating gametes or sex cells. If you have trouble remembering what a gamete is, I always tell my students, gametes are gonna meet, okay? Gametes are gonna meet. So the sperm and egg are gonna meet to make your 2N organism eventually. But to make a gamete, we have to actually change the ploidy and go from diploid to haploid. So we need we only need one set of the chromosome for each gamete, because if you didn't do that, you would all of a sudden have some polyploidy going on. And that can cause all sorts of things like speciation, which we can talk about during evolution. Another big thing about meiosis is that we're creating genetically unique cells. So unique meaning the actual distribution of genes and the variety of alleles are different from the parent cell, which is really important. And we'll talk about that more with evolution as well. And, why is genetic variation so important? And we talk about diversity as well, even as far as like an ecosystem goes and how important diversity is in terms of being able to handle changes in the environment. Okay, so here's a very, hand-drawn diagram of meiosis. What I want you to focus in on here on this image is, here's my laser pointer. Notice right here after meiosis 1 that we have already become haploid. And a lot of students get that mixed up because they think, well, how can you be haploid at the end of meiosis 1 and then I have a whole other division going on? So make sure you know that the actual division of going from diploid to haploid happens during meiosis 1. Okay, the reason that is that if you look at the very beginning of meiosis, you have homologous chromosomes together that are going to be separated. And I have another picture that'll show your homologous chromosomes a little bit better. So if you are kind of freaking out about homologous chromosomes, don't worry, because I will show you a picture, I promise. Okay, let me just think real quick. And then turn up my mic to be a little louder. Sorry, everybody. Thank you so much for being patient. Okay, so homologous chromosomes, right? So that is where in the beginning of meiosis 1, so think about how we have chromosomes 1 through 23, okay? You have a copy of chromosome 1 from mom and a copy of chromosome 1 from dad. And so prior to meiosis, we're going to have that DNA duplicated. And so each chromosome is going to have its own identical sister chromatid. And in meiosis 1, our job is to separate those homologous chromosomes. Once we do that, we have gotten rid of that diploidy. We are now haploid. And the picture on the next slide will be a little bit better for this. So let me just move over. So when we talk about your homologous pair, right, we've got a pair that's from mom and a pair that's from dad. These two sister chromatids are identical prior to the crossing over. So moving on to where we get some variation, that's happening during prophase 1. Okay, so during prophase 1, the homologous chromosomes line up and they form your synapses. And if you don't know that vocab word, I wouldn't worry about it too much. And they actually cross over and exchange genetic material. So they recombine. And so maybe if this is the gene for dimples, and let's say mom didn't have it and dad does have the allele for dimples, now all of a sudden we have this variety of genes, right? So maybe mom, this chromosome now, right, originally didn't have the gene for dimples, and maybe there's other alleles here, but now we're just kind of mixing it up. And so we get a huge genetic variation going on right there. Another big way that you need to know that gives us genetic variation through meiosis is independent assortment. So independent assortment, this image is nice because it shows a variety. And this is just, an organism with 3 sets of chromosomes, or not 3 sets, 3 chromosomes. And you can see here how like all the blue are lining up on the left, all the red on the right. Over here, it's red, blue, red, blue. So how those align they align independently from all the other ones, right? So there is no rhyme or reason to which homologous pair aligns on the left and which aligns on the right. So there are 2 raised to the 23rd possible combinations for us. right? So those are a huge variety of combinations. So not only do we have each homologous pair participating in crossing over, but we also have independent assortment going on. So that also creates huge genetic variation. Looking at this in terms of asexually reproducing organisms, would an asexual reproducing organism ever participate in this? It would not, right? Because they're just using mitosis to reproduce. So they don't need to worry about meiosis at all. If we think about mistakes, this is a huge theme in AP Bio that they call it like a disruption to the system, or, you know, similar words like that. But the idea of like, what happens if this process is disrupted? And you need to be able to make a prediction of, okay, well, where did that disruption happen? What was likely the cause, and what likely is the outcome? So here, if we have our microtubules that are extending here, and we have microtubules that attach to the centromere here, but then we also have ones that do not attach, and those kind of help lengthen the actual size of the cell as it's starting to separate. But if we have one of these fibers break, that's called non-disjunction. That is something you should know, non-disjunction. So they become disjointed, they get separated. And what happens there is that when this is a picture during meiosis 2 here, because and the reason you know that, by the way, is that your homologous pairs are on top of each other. They're not next to each other. So you have like a sister chromatids are being pulled apart here. So anyways, if this, for example, were to break, that spindle fiber was to break, then these sister chromatids would not separate, and meiosis would continue, and you would have daughter cells that were missing a chromosome, and then another set of daughter cells that had too many. And so here's an example of what might be the outcome of that, right? So here we get a trisomy of the 18th chromosome. So trisomy meaning there's 3 copies. And a lot of trisomies that happen actually end up, it would never create a viable organism. But we know there's trisomy 18, trisomy 21, Down syndrome. And so there are some trisomies that aren't fatal. All right, if we also talk about variation, you know, with meiosis, but also just the big picture of why variation happened. I told you I was going to talk to you about this in ecology and evolution, but I'm throwing it in here too. So in evolution, when we talk about bottleneck, the bottleneck effect, right? How we have this existing population right here, and they go through this bottleneck event. So for whatever reason, they get, you know, a couple organisms from the population get separated, or maybe everyone dies but them because of some catastrophic event, right? Because maybe there's some hazardous weather or whatever. It bottlenecks the population, which means only a few from that original population get through and survive. And so as you can see here, the original population had all sorts of different allele variations of whatever trait we're talking about, right? This is just a model. This is not, you know, an actual specific organism we're talking about, but we're looking at all this variety of all these alleles. And now the bottleneck event happens and you only have 3. So this population ends up looking like the original founders, right? That founder effect. The reason why this matters is that if you think about variation, and when you lose that variety, like let's say this one right here, this light green color, the white and the black, let's say those variations were really important for organisms, and they just so happen to be able to withstand drought very well, right? So maybe we're looking at a plant or something. And if those variations were lost, and now this new population experiences a drought, and none of them have that natural capability of withstanding a drought, then your entire population might be gone. Okay, so without that variation, you're very unlikely, or not unlikely, it just makes you unstable, and it's difficult for you to handle changes to your environment. So again, there's that theme of disruptions, right? So like if your environment is disrupted, or, you know, something changes drastically, without that variation, you're little, and you don't even have to be little in size. You can be a giant population of 2,000 wildebeests, right? But if you don't have much variation, something might come through and knock everyone out. Okay, good. Now let's talk about Mendel and meiosis. So we go through meiosis. We have you look at, you know, these two separate division events. We have segregation happening, which means your actual, you know, if someone's big R, little r here, those 2 alleles have to separate, right? So mom can't give both alleles to each child, only one, right? And same thing with dad. So when we split them up, that's where the Punnett square gets built. That's why when you do this Punnett square, you can't do big R, little r next to each other and then add the y's. So you have to split them up. So that's law of segregation. And then independent assortment is where you get, I kind of tell students, it's kind of like foiling in math, which don't hate me for saying foiling. But remember in math, when you're doing foiling and you have like, you know, I would say, hey, underline the big R, put an arrow to the big Y. That's one option. Go back to your big R, put an arrow to the Y. So you've kind of foiled your R to your other Y options. And that's kind of how I help my students figure out how to build these larger Punnett squares. Anything larger than this, so if you are given a problem where there are 3 or 4 traits that are being crossed, do not make a giant Punnett square. Okay, you don't need to do that. You can use the law of probability instead. And this is where, let's see if I can do this. I'm going to go to a different screen and then see if I can share it with you, because I think this is really important. So I'm going to see if I can share a different screen and then write, because that would just make my day. I know you can't see my face, but I'm going to see if it will let me share a different screen. So I'm going to stop sharing for just a second. And then I'm going to try to share a new screen. Let's see if it will let me. Oh, it might. Okay, I hope this works. I really, really do. Okay, so I'm hoping this works. And if it doesn't, I'm sure somebody will ping me and tell me, knock it off, go back to the PowerPoint. But anyways, what I was trying to tell you is that if you're given a huge problem like this, where, you know, they're giving you some giant cross, right? Don't sit there and worry about, oh my gosh, I don't have time for this giant Punnett square. What am I going to do, right? Because maybe they say, hey, the parent generation is heterozygous for all 4 traits. My goodness, all 4 traits. And they want to know, what's the probability of getting a, let's say, homozygous for all 4 traits. Let's hope this works. Oh no, I'm getting pinged. That means it's probably not working. Oh no. Sorry, everybody. Okay, notebook looks great. Thank you, Peter. Okay. Oh, this makes me so happy. I was very worried that there was no way I could write. And I love writing for you all. Anyway, so if they say, hey, what's the possibility of this? Don't do some giant Punnett square, right? All you need to do, we can assume these are all going to segregate. So what I would do, and you don't have to actually, because once I do this, you'll get the idea and you'll be able to do it in your head. But separate each of these traits and realize, hey, look, for each of these traits, there's going to be a 1 in 4 chance for that homozygous recessive. Okay, so I could do it again for B, I could do it for C, but again, I think once you see this, you'll realize how you can use the law of probability to figure this question out. So you would do 1/4 times 1/4 times 1/4 times 1/4 to get your answer. So 1 over, was it 64? So anyway, that would be a really easy, quick way to answer a question that has to do with Punnett square crosses, but you're not actually necessarily making a big Punnett square, and you're able to use laws of probability instead. So that's another math skill that you are responsible for. So let's go back to our presentation. And let's reshare. Sorry, you don't need me to be talking you through all this. Sorry, everybody. Okay, so Mendelian genetics, making sure you know what it is, right? So Mendelian genetics basically just means, hey, you know what? Our alleles are going to segregate, and maybe nothing's linked. Nothing funny is going on. We don't have codominance. It's not sex-linked. It's on the autosomes, which means it's on the chromosomes 1 through 22 in humans. We talk about autosomal dominance. I said the word autosomal has to do with non-sex chromosomes. So your autosomes are chromosomes 1 through 22. Dominance just has to do with, hey, if they tell you it's dominant, that means that the only thing that's going to show the recessive trait is going to have to be homozygous recessive, right? Because if you have the heterozygote, that one dominant allele is going to shine through. Okay, this is an example of a dihybrid cross because we're talking about two traits, dihybrid, and here you get a 9:3 to 3:1 ratio. So I'd be familiar with these ratios only because you may be asked to do a chi-square, for example, and I will do a sample chi-square, so don't worry, that's coming next because I had heard from our awesome TA, Jed. Shout out to Jed, by the way. I know you guys are all interacting with him on Discord and in the chats and the questions and everything. And anyways, so Jed let me know before tonight that everyone was asking for a chi-square. So we will do a chi-square in a minute. So anyway, the reason why it's good to know your expected ratios, right? So if they say, hey, we've got this dihybrid cross, both parents are heterozygous for both traits, you know, and here's our outcome. Look at, there's like so many that are all, you know, big R, little, or you wouldn't know the genotype, but they'd say, hey, there's so many that are yellow, right? And we wouldn't expect that many to be yellow. Is something going on or not? And so you would use this ratio that you know to serve as your expected in the chi-square test. Okay, so here, this is not a chi-square, but we're going to do this one first, and then we'll do chi-square together. So this is just a quick practice problem. Pea pod color inherited through simple autosomal dominance. So green variation is dominant to yellow. If the cross of a green and a yellow plant create offspring ratios of 13 green and 15 yellow, what were the parent genotypes? So I'm going to give you a minute to do that. Sit there. Okay, so your first indication here, I had students see this problem before, and they're like, wow, 13 is way less than 15. But really, if you think about it, if we add those together to get your total population, it's 28. And 13 out of 28 and 15 out of 28 is really close to 50% each, right? So really, you're getting 50-50 of your two phenotypes, okay? So we've got a green plant and a yellow plant. So each parent must have at least one recessive allele, because if you do have any yellow plants, that means each one must have at least one, okay? And then the other parent, right? Or sorry, I'm getting too confusing here for you. I'm sorry. So if the yellow parent, you know for sure, is little g, because it told you that green is dominant to yellow. So you know one parent. You figure out the other parent must be heterozygous because they do have yellow offspring. If you have recessive offspring, then both parents have to have at least one allele. And so here's just a picture of that cross. But again, that's pretty simple. So I'm going to pop back over to do the chi-square question. And then after that, I'm going to give you guys a quick, like on your own multiple choice practice through this cool program called Mentimeter. And so you just do it on your phones and it's self-paced. So, and it'll tell you the answer right away. So when you do that, you'll take your little break and then I will let you know when we do that. So just give me a moment. Okie dokie. Stop sharing, share screen. Window. Here it is. Here we go. Okay. All right, so we're talking about chi-square. Chi-square is a statistical test to help us see whether or not the stuff that we're seeing is due to chance or maybe something else is going on. So we see it a lot in genetics. Hold on one second, make sure we're good. We see it a lot in genetics. And it's really similar to how, you know, when you look at error bars and you have to use the error bars to say whether or not what you're seeing is statistically significant, chi-square is very similar. So if I just gave you a quick sample problem, let's say we'll do the yellow and green thing again, okay? So we cross yellow and green. Sorry, my brain is hurting right now. I'm going to do a different problem, aren't I? No. Okay. So let's say we said green is dominant, right? And we know yellow is recessive. And we cross what you can call like a true breeding yellow, which means it's going to be little g, and a heterozygous green. Okay, so if we did that, just like I showed you before, you expect 50-50, okay? Wait, hold on. My brain is hurting, 1 second. Sorry. Little g. Sorry, I'm going to make better handwriting. So anyways, you expect 50-50% ratio. Okay. Let's say a farmer actually does this and they realize, hey, actually, my observed is I have 69 that were yellow and I have 31 that are green. All right. You will not be given a table like this, which is one of the bigger things that, actually, I can't say never. Maybe they'll give you a table. Maybe they'll be super kind to you. I don't know. But my students always would get to a test and I wouldn't give them the table to organize their information. And they'd be like, I didn't know where to put anything. And so be prepared to organize your own thing, right? So like your categories are here on the left, right? So our only categories here are yellow and green peas. We observe that 69 were yellow, 31 were green, okay? Your total there is 100, right? See how that was kind of an easy math thing for you? So if we look at our expected, if we have 100 total, that means we would expect 50, right? So we get 50-50 here. So you do the math where it comes from here, right? So you need observed minus expected squared divided by expected. So observed minus expected squared divided by expected. Here is where students mess up the math because remember order of operations, remember that whole PEMDAS thing from when you were in elementary school? Okay, if you just throw that in your calculator is going to do that exponent first, and then it's going to do the multiplication, then the division, then the addition and subtraction. So the subtraction is going to happen last, and we need that subtraction to happen first. Okay, so use parentheses, be smart with your calculator, and just double check your work. Okay. So anyway, you do that. So your observed minus expected is 69 minus 50 divided by 50, and make sure you square that 69 minus 50. I'm just going to quickly throw in the answers. We'll do another one together where I put you on your own a little bit. Okay, so don't worry. Anyway, so 0.48 for green. Our value is 1.44. And so we go back to the equation and it wants us to sum that. That's what that sigma is. We want to sum it to get the chi-square value. So the sum of those two numbers is 1.92. So that is my chi-square value. My chi-square value is 1.92. So now what do you do with that, right? Again, let's zoom out and realize, okay, we're trying to figure out, we know that if we cross these two plants, I should expect 50-50. Right? But I didn't get 50-50. I got 69-31. But also we know tossing a coin, you have a 50-50 chance for either side, heads or tails. But you've probably done this in class, right? Where you do this and you do it 100 times and there's always that one lab group that's like, I got heads 80 times. Oh my gosh, right? But that's actually kind of nice because then we can throw in chi-square and figure out, well, do they have a trick coin or were they cheating? Like what happened? Or is it just due to chance? So again, we're trying to figure out, is the variation we see in the observed population, is that due to something else other than chance? So we use this chi-square value, and what you're going to do is that chi-square value is going to fall somewhere on this table, okay? These numbers here are called critical values. I don't think it really matters that you know that name, but anyways, critical values. And we usually look at a p-value of 0.05. Right? If you're doing like pharmacy work or creating, pharmaceuticals that can, or for people and you don't want to hurt anyone, 0.01 is preferred, but 0.05 is okay when we're looking at biology and genetics and we're not hurting anybody. So 0.05, and I'll explain what that 0.05 means in a minute. Anyways, you want to see where does this 1.92 land? And then to figure out where you're at, you need to know your degrees of freedom. Your degrees of freedom is n, minus 1. So n meaning the number of categories you have. So in our case, 2 minus 1 is 1. So our degree of freedom is going to be 1. So you find your degree of freedom, it's 1, and you say, okay, well, where does my chi-square fall on that table? Is it bigger than this critical value or is it smaller? So we have this value of 1.92. It's not bigger. It's definitely smaller, right? So 1.92 is less than our critical value of 8.4. So it's a really small difference between what you observed and what you expected. And so when we go to the left, if you are to the left of your critical value, we fail to reject the null. And I know I'm jumping ahead, so don't freak out, okay? Promise. So we fail to reject the null, okay? What the null hypothesis is, the null hypothesis says that there is no significant difference or relationship. So it kind of, I'm stating it kind of vaguely because it really depends on what we're talking about here. Like there's no perfect definition, but basically the null hypothesis, null sounds like no. So it means like, hey, there's nothing funny going on here, right? What we're seeing is just due to chance. It's just due to chance, okay? So if these numbers were bigger, right? So if our chi-square value was big, you know, like let's say it was 9, so it was in this direction now, right? If it was bigger, that means that our observed values are super, super far off from our expected. So something else must be going on. And so here we are able, if we go to the right of our critical value, we can reject the null, which, by the way, if you're rejecting the null, that means you get to accept your alternative hypothesis. If you don't know what an alternative hypothesis means, it's basically, you know, think about when you learned about a hypothesis for the very first time, like maybe second grade, you know, your hypothesis was like, oh, if I change this, then this will happen. So your alternative hypothesis is that, right? The alternative hypothesis is the one that says, hey, you know what? There is something significant going on here. There is maybe an alternative method of inheritance going on. Maybe this isn't typical Mendelian genetics. Maybe this particular trait is actually sex-linked, right? And we just never knew. And now all this data over and over again, we keep running the same experiment and we keep getting this huge chi-square number. So we can reject the null and accept your alternative hypothesis. Okay. Phew. So we are 30 minutes in. So ideally, I'd like to be moving on to the next unit. So one moment. Dun, 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 dun. So I'm going to go very fast through some non-Mendelian genetics, just so we figure it out, or just so we get there. Be sure that you're able to make a Punnett square using your sex-linked information. So for example, we talk about sex-linked. That means that the trait is on the X chromosome most of the time. And I'm going to pause really quick, actually, because we're getting some questions and I want to make sure we answer them. How did I get 0.48 and 1.44? Do we not get the squares? Okay, one second. Just make sure my math is right. Some people are having some questions about the chi-square. I just want to make sure we are good. Oh, I know why. Let me go back. Sorry, everybody. You're right. Dun, dun, dun. This is why making math, the mistakes, happens to teachers too. So, oh my gosh, I'm so sorry. Let me get my window open. Thank you, Jed. Thank you, people. So what I was doing, I was working on a problem earlier that I was going to show you where I was giving you 75-25. Not 50-50. So that's where my numbers are coming from. I'm super, super sorry. So my numbers are coming from what I had originally planned on doing, which was where we would see 75% green and 25% yellow. Okay, so that's where my numbers are coming from. So if we redo this, and let's just keep it with my 50-50, that's fine. And this is why you are all so wonderful. Thank you. This is what happens when I'm trying to come up with example problems and I have little children in my house trying to talk to me. So again, I apologize for that. So we'll keep it with the 50-50. And instead, so sorry about that. Good, good, good. Thank you, Jed, for catching that. Thank you for all the participants who are here and chatting. I really do appreciate that. Okay, so I don't have my fancy calculator, which is why I tried doing the math ahead of time. Okay, so if we do 69 minus 50, sorry, everybody. I'm using my phone calculator, of course. You get 19, right? So you're going to square 19 and then divide that by 50. Yeah, good. So your first one should be 7.22. I'm going to write it in red. 7.22. Okay, we do the other one. So you're going to do 31 minus 50. You're going to square that. Is my calculator right? I'm getting 7.22 for both. Okay. So anyways, I am getting 7.2 for both. So when we add those together, we get a huge number. We get 14. And so then we would be rejecting the null. Sorry about that. We'll do another practice problem to make it a little bit better. Alrighty. Okay, so let's do, I will come back to this one actually. We'll do a practice problem later, I promise, to clear it all up. But for time reasons, I do want to keep moving on to all the other things because I don't want to get to a spot where we don't even have time for ecology. So if you were given a sex-linked practice problem where the father has hemophilia, and it would tell you, hey, this is a sex-linked recessive disorder. So if the father has it and it's a recessive disorder, males are XY. So that means he has the recessive allele. And they tell you that the mom is normal for blood clotting. Okay. All the information they would give you on difficult questions about some disease or some drug or something that you've never heard of before, they'll give you the background information. So everything you need to answer the question will be there. So don't worry when you see some information that you've never ever seen before. And in fact, expect it. Expect them to give you some 2 or 3 paragraph explanation before you attempt a problem. So the probability that their son has hemophilia, be careful about that too, where you're looking at son options, right? 100% would have it. Oh, wait, sorry. Yes. No, it's 100% would not have it. Yikes. Sorry. My brain is a little messy right now because my system wasn't working earlier. So I kind of got all jumbled up. Okay, I can move through it. So if he does not have the recessive allele, 100% of the males will not have it. The only way the male will have this recessive disorder is if the mom is a carrier or if the mom has it. Okay, let's look at pedigrees and inheritance. I know students struggle with this. So if you've never seen a pedigree before, circles represent females, squares represent males. When you have a horizontal line, that's representing 2 people that are having kids, right? So then did I say horizontal line? Then the vertical line represents all their children. And then notice how like person 4 is over here, right? So they have, I guess I used to say married for my students, but that doesn't really matter. So they're going to mate with person 3, and those are their children right here. And so when you look at this and you're like, okay, well, what kind of inheritance could this be? I have no idea. Try to figure out, like I always like to cross off sex-linked first, right? So for example, if this was sex-linked, that means this mom has it. And if she has whatever we're tracking, both of her sons would for sure have it, right? So right away, I know it's not that. Then I can also look at this and say, okay, well, if this is a dominant disease, right? If I'm going to guess autosomal dominant, I know that's not true either, because this dad right here would have to be homozygous recessive. And if it's a dominant disease, then more of these children would have it. You wouldn't really see this skipping of generations, right? Even if, yeah, this person could be heterozygous and they could have a 1 in 4 chance of having, their children be unaffected, but that would just be really unlikely. So if we're looking at something like this, this would be, autosomal recessive. So with recessive, you tend to see it be less apparent and also potentially skipping generations. Okay, so we're skipping generations here. If we're looking at something like this, you're like, oh man, okay, well, the female's affected and the males have it as well. So you could think it's maybe sex linked, but here's something that jumps out to me. This female who has it, all of her kids have it. This female who has it, all of her kids have it. And so this is an interesting one where something that maybe was like brushed over in class, but the mitochondria has its own DNA, okay? Sperm cells do not have mitochondria. Egg cells do. So when the fusion of the sperm and egg happen, and then that entire organism comes from those genes, from what was in those cells, come from the original mitochondria in the egg. So all of our mitochondria have DNA from our mothers. So if there is some trait that's being passed down from the mitochondria, if the mom is affected by it, all the kids will be as well. So this would be a mitochondrial inheritance. Okay, gene linkage is when we say rules don't apply necessarily, right? So when we normally expect segregation of traits or alleles, it doesn't happen. And so the fruit fly has been a really good example of this because they have a lot of genes on the same chromosome that are very close together. And these numbers are representing a relative either close together or not, right? So from 0 over to 104.5, those would be like map units. So we're mapping out how far these genes are from one another. So the closer they are, the smaller the number. We can also call that a recombination frequency. So the recombination frequency is how frequent is the recombination of genes, which is the crossing over during prophase 1. So if it's small, then they don't recombine very often because they're so close together. The further they are, the easier it is for them to mix. If we're less than 50%, we usually say they are linked. So that means they're on the same chromosome. And so when we're mapping out a chromosome, we calculate this map distance, but that doesn't necessarily mean it's like a very specific nucleotide distance. It's a relative map. So just be careful of that. Okay, so quick sample problem. Go ahead and take a look at that. Okay, so when you get something like this, I like to draw a line first, right? And then I choose the two genes that are furthest apart, two alleles that are furthest apart. So J and M are 12 apart, right? So the recombination frequency is 12. So they're the furthest. Then I kind of just start problem solving and I throw one on there and I think, hey, well, okay, I know that L and M are 7 apart. And L and K, or it's not L and K, sorry. L and J are 5 apart, or that's what makes them. And then you kind of just process of elimination, right? And see if it works. With K, you could try it in between J and L, or you could try it beyond J. Eventually, you come to it that J and K are 3 apart, and then L and K are 8 apart right here. And so that's what makes the most sense. And so your order would be K, J, L, M. Okay, I know I went through that very quick. If you're like, what the heck is she talking about? My teacher never even covered this. Honestly, there might be one question about this, right? And that's also something I want to encourage you all on, is that if there's one topic that like you do not know very well, like one thing, and you're like, I'm never going to understand meiosis. That's okay, okay? The exam is tomorrow. You have done everything you can. You are here and reviewing everything you've learned. So give yourself some grace and realize that if there's a topic you don't know, it's not going to be every question, right? It's going to be 1 or 2 questions. And if you don't know it, try your best and move on to the next one and get the ones that you know right. All right, so take a deep breath. It's all going to be all right. Okay, so I'm going to skip this fruit fly example because we already did an example, so we're good to go. So moving on to DNA replication, protein synthesis, regulation, mutations, biotech. We're going to try to hammer this all out so we have time for evolution and ecology. So big things about DNA replication that gets mixed up with transcription and translation is that everyone likes to think about DNA replication within protein synthesis. But remember, DNA replication is happening during the cell cycle. Okay, so we're making an exact copy of DNA. It needs to first be split. So like Anne said, those hydrogen bonds get opened up down the middle right here. And you've got all sorts of enzymes and proteins who play a role in this. This is not just one amazing enzyme that can just do all the jobs. You've got a bunch of different enzymes involved. You need RNA primer put on first. That's what this red is representing here. So RNA primer has to get put on first before DNA polymerase can come in and synthesize nucleic acids or nucleotides. And like Anne said too, pay attention to this directionality. So amino acids have directionality and so do DNA. So does DNA. So notice how the new strand is being synthesized in the 5 to 3' direction, right? Which means it's reading the template strand in the 3 to 5. Okay, so it synthesizes in 5 to 3. The same thing is happening here on your lagging strand, right? This first one is your leading strand because it's just following the enzyme called helicase down here that's unzipping your double-stranded DNA. So DNA polymerase is just following helicase, and it can just add nucleotides in this really pretty nice strand, where on the lagging strand, DNA polymerase has to go in the 5 to 3' direction. So it goes in the 5 to 3', and then it has to backtrack, right? And then synthesize more DNA, and then backtrack and synthesize. So you get these lagging strands here that end up obviously getting connected, and DNA ligase, there's another DNA polymerase that helps to do that, but that's beyond the scope of the course. So just knowing that DNA polymerase is your enzyme that's synthesizing the DNA, so adding nucleotides that are complementary to the template, and then which is you've got a leading strand here, lagging strand there, and that they're both going in the 5 to 3' direction. Like I said before, disruptions are a huge theme. So think about how could this be disrupted if one enzyme is altered by some environmental change, or maybe, a T is brought in instead of an A, right? Or sorry, a T is brought in instead of a G, right? So this C is supposed to be bound to a G, but instead we have T's. Or, sorry, I don't need to go into all the different explanations, but obviously that's going to hinder your DNA replication. And most likely and hopefully that cell is going to be told to actually undergo apoptosis. Because if the mistake is big enough and it's not fixable, which proofreading does happen in DNA replication, by the way. So they do proofread and the enzymes proofread and can fix it. But if it's not fixable, that cell should just go away and do program cell death. One more quick thing about 5 to 3'. It all comes from this directionality of the molecule. So the phosphate right here is going to be on the 5' end, which means it just basically says that this fifth carbon on your sugar is connected to the phosphate, and the third is connected down here. Okay, so we have 1, 2, 3, 4, 5. So again, we have carbon 1, 2, 3, 4, 5. So that means a new nucleotide here is going to have its 5' end connect, which is already starting, right? It's 5' end connecting to the 3' end. So 5 to 3'. Okay, I'm going to move on from these questions. Protein synthesis. There's so many ways to say protein synthesis. It's also called gene expression, also called central dogma, also called transcription translation. So there's tons of things that mean the same thing. So I know you're like, okay, why does biology do this to me? But anyways, so protein synthesis is when we are transcribing DNA to make mRNA, and then that mRNA is going to be translated at the ribosome to make your protein. Okay. In the most general sense, we've got these 3 steps. I wouldn't necessarily think of them as steps, but get the idea that like, hey, it has to start somewhere. So the big enzyme involved here is RNA polymerase. RNA polymerase is like the MVP because it doesn't need any other helpers. It doesn't need like helicase or RNA primers or anything like that. It can just synthesize. It's wonderful. It recognizes the promoter, which is the beginning of the gene, and then it's going to go ahead and read the template strand, which is also known as the non-coding strand, which I know sounds really crazy that like the thing that it's reading and making nucleotides based off of is actually called the non-coding strand, but that just means the one that's not being synthesized is your coding strand. The reason why is that your RNA that's released is letter for letter or base for base identical to the coding strand. So RNA polymerase grabs on, it elongates, making your nice pre-mRNA molecule, and then it reaches the terminator sequence and comes off. If we're talking about prokaryotic cells or eukaryotic, what happens next is different. So in a eukaryotic cell, you're going to have post-transcriptional modification. So you get your addition of your 5' cap, you get your addition of your poly A tail, and your removal of the introns. Okay, this is not going to happen in prokaryotic cells, but it does happen in eukarya. So again, that's another big theme that you may be asked about, and you might think it's coming out of left field because you weren't explicitly taught something about, you know, prokarya versus eukarya in that topic. But again, go back to your basic knowledge. Even if you have to jot it down on a piece of paper and be like, okay, what do I know about prokarya? What do I know about eukarya? Okay, I know that in prokarya I have no nucleus. I know that transcription and translation are happening at the same time. So take some time on your test to annotate, draw stuff out, write down stuff you know real quick. Okay, if you get to a problem and your brain starts to spin, close your eyes, take a minute. Okay, take 30 seconds. Open your eyes and draw something you know. Write words that you do know, and then go back to the question. Okay, if the question is driving you like to a panic attack, skip it. Go to something else that will enlighten you and make you feel like, oh, I know this one. Okay, good. Right? Obviously, if you're taking the digital exam, that looks a little different in terms of being able to go backwards or forwards. So I apologize for that. But for those of you taking it on paper tomorrow, use that. Okay, use that strategy of writing on paper, drawing something out. Those of you taking it digitally, you can still draw out. You still have paper. You can still put paper in front of you. Okay, so we're going to take a break in just a second, I promise. So with translation then, obviously, this is something that you learn in 1 week in school. And so this is a 5-hour cram. So keep in mind that I can't talk to you about every single thing. But what you should know is that this is happening at the ribosome, that your mature mRNA is going to show up and be read, okay? And it's going to synthesize protein. So what happens is that you've got your ribosome, or your mRNA is going to be read in this triplet code, right? So it usually starts with always starts with AUG, okay? So AUG is your codon. tRNA is going to have the anticodon for that, so the complementary pairs to that. tRNA will be bringing with the correct amino acid, okay? Your ribosome is going to move along the mRNA and shift. So with every shift, you get a tRNA that leaves and a tRNA that comes. And through that shift, and we've got enzymes going on here too, but we get this covalent peptide bond between your amino acids, okay? If I have more time, I will go back to this, but I want to just keep moving. So make sure we know where transcription occurs in prokaryotic cells and how can we take advantage of this. Okay, I'm going to come back to gene cloning. So we will do that next. I'm going to take a quick break, and while I'm doing that, I'm going to have you all do this quick little game. So hold on. Well, it's kind of a game, I guess. It is what you want to call it. Okay, so I'm going to stop my sharing and then share again. And then let's see if this will work. Chrome 10, maybe? Mentimeter. Here we go. Okay. Nope, wrong one. Okay, so shoot. Sorry, guys. I wish I was like the best at technology, but sometimes I'm just not, and I feel bad. Okay, maybe if I do my entire screen, and I won't do that again, but okay. So here's a QR code. Go ahead and take a picture of it with your phone, and it should pop out to this Mentimeter website where you're going through a bunch of questions by yourself. So take a moment to do that. And I'm going to ask Jed if it's working. And hopefully it works. Okay, so if the QR code does not work, I will try to fix it. But in the meantime, I mean, it's showing up on my screen, so. All right, let's see. Hopefully it works. This is why I miss the chat. I miss being able to see your guys' chats. Works for Jed. Awesome. Okay, so I'm going to take a quick break to have some water. Good. It's showing a meiosis and mitosis question. Good. Awesome. So work through these, everybody. And I will be back. It's about 16 questions. When you're done, you should take a break as well, okay? Oh, yay, and it's already working. People are already answering. Wow, you guys are so fast. Okay, so take a break. I'll be right back. Sorry. I got all some clothes. Okay, everybody, I'm back. Sorry, I hopped onto Discord and saw some people wondering, like, what happened? Is my connection out? So I'm here. Sorry. Just wanted to take a quick bathroom water snack break, which I know you guys are excellent and very experienced crammers, but we had to move on. All right. So real quick, I just wanted to talk about gene cloning because you will be responsible for knowing about it. So we think about taking advantage of bacteria. Okay. And I know this picture has a lot in it. So what I might do is actually even hop over to my drawing. I tend to prefer drawing things out for people because sometimes a good old handwriting is very, very helpful. So let me get that window open. Okay. Nope. Wrong one. Sorry. Man, I'm like not doing my best tonight. Sorry, guys. Telling you. Window. There we go. Here we go. Okay. So gene technology, if I have, we know bacteria have circular DNA. Okay. We can take advantage of those circular DNA called plasmids, and we can insert genes into them. So big things you need to know. Our end result of this recombinant DNA technology or, transformation, there's lots of different ways you want to call it, or gene cloning, okay? Your end result is that we have this original bacterial cell that now has a certain gene that we want, right? So it has we call it like a gene of interest, right? It's the gene we want it to express. Bacteria are really good at this because, first of all, once we get a certain gene of interest inside, they will go ahead and transcribe and translate, and they have no idea, right? This is huge in the medical field, right? We used to get insulin from other animals. Instead, we inserted the gene for insulin into a plasmid, and now bacteria can make it, and it's making human insulin. It's incredible. Okay, so big things you need to know about this is, first of all, restriction enzymes. So restriction enzymes are able to make a cut, right? So it's able to cut, it's able to cut the DNA at specific sites. So we've got very specific sites that it's going to cut them. You want to cut your plasmid one time. And I have a lot of students who are always like, well, you need sticky ends on both sides. Why can't you cut it twice? It's a circular piece of DNA. So cutting it once gives you your two pieces. All right. If I have my gene of interest, right, here's my gene of interest. And again, I'm drawing this in a very cartoon-like way. Here's my gene. Say it's only that section that I want. And here's the whole DNA. We need to cut outside the gene twice, right? So I get one cut on the plasmid, two cuts on the gene. You don't want to cut inside the gene, so you have to choose a correct restriction enzyme. But what the restriction enzyme ends up doing is creating sticky ends on both your plasmid and your gene. So it creates sticky ends on both so that it can fit in there. And again, I'm going to try my best to draw a little bit. And for those of you who are asking about the multiple choice questions, we will go over those, so don't worry. All right, so we get a sticky end here. And again, this is a cartoonized copy. I'm going to draw two layers here to show we've got a double-stranded piece of DNA. So the sticky end is going to be, like, notice here, right? There's nothing there. And so you have your bases that have no one to bind with. So they're pretty upset about that, right? And so they want to make hydrogen bonds with other bases. So you have sticky ends on both sides of the plasmid, and then you would have the sticky end on the gene. Okay, so sticky end, and DNA ligase is going to come in here and form that covalent bond so that it's really sealed in there nice. I would say those are the big things you should know for gene cloning. So restriction enzymes cut at specific sites. You need to cut the plasmid once, the gene twice. And then obviously this has to go back into the bacteria. Okay, I don't believe you need to know the specific steps, but you do need to know it needs to go back into the bacteria. Other things that might come up here is that we may also choose plasmids that already have, the gene for let's say you have the gene for amoxicillin resistance, right? So amoxicillin is an antibiotic. And so if you have the gene to be resistant to it, that means if you are grown on a plate with amoxicillin, you will be fine. Okay, so we use that as your positive controls as well, just to make sure that like, hey, you know, if we do this right, I know I have my gene of interest and my amoxicillin resistant gene, and you would grow it on amoxicillin and you'd grow it not on amoxicillin too. And so, and some people do this too, not with just amoxicillin. They might put like the jellyfish gene in there. And so if it glows, then they know transformation has been successful. But those are some things that might come up. And I can't tell you exactly what they would tell you because every experiment will be different. You'll just have to kind of synthesize and look through to figure out what's the control, what's the experimental. But again, yeah, that's put in there so that we can be sure transformation has occurred. Because otherwise, if you have bacteria growing on a plate, we can't see microscopically into the bacteria to know whether or not transformation has been successful. So you can use amoxicillin or you can use presence or absence of the jellyfish gene. I'm sure there's other ways you can do it too. So don't quote me on those being the only things. Okay. Real quick, let's go to our questions that people were having about the Mentimeter. So let's go here. Presenter. Okay, so which part of meiosis is most like mitosis? So most of you got that right. And the reason, oh no, wait a minute. Sorry, most of you did not get that right. That's why you guys want to go over this. My bad. Okay, sorry, everyone. Oh, look, somebody changed their answer. You guys are funny. Okay, so when we think about mitosis, the biggest thing that I think of mitosis in terms of chromosomes is that when you have your division, your sister chromatids are dividing, or they're splitting, right? Your sister chromatids are splitting. And so in meiosis, your sister chromatids don't split until meiosis 2. Okay, so it's most like meiosis. Meiosis 2 is most like mitosis because that's where you have your sister chromatids splitting. So again, these questions I typed up, they're very short, they're very quick. So if you got one or a couple wrong, you know, it's not the biggest deal in the world. Okay, what is analogous to an identical twin or a clone? So for this one, again, maybe I just wrote a bad question. But when I think of identical twins, I was trying to get you thinking about the sister chromatids because your sister chromatids are the identical copy of that original chromosome. Okay, the homolog is going to be different. That'd be analogous to like you're like a not identical twin, fraternal twin, right? So yeah, you've got a lot of the same things, but your variety of genes are much different. And I can draw a picture of that if that would help real quick, because these two questions are making me think I need to talk about that, and it's worth talking about for sure. Chrome tab. No. Sorry, everybody. I do want to draw this out for you because I think it's important. I've said that a bunch of times tonight, haven't I? Like, oh, I think it's important. Okay, so when we have DNA, normally during interphase, it's just chromatin, right? It's not until prophase that it condenses into your chromosome, okay? So remember, during interphase, this duplicates, right? Because interphase is G1, S, and G2. So S is when your multiplication of DNA happens, your duplication happens. So when we show a sister chromatid, I drew this poorly. This looks bad. It's supposed to be identical. We draw a sister chromatid. I like to think of like, oh, well, this one is the original, and this one is the identical copy that was made during S phase. Okay, so when I say identical twin, that's the one I'm talking about. I'm talking about the sister chromatids, right? So these are sisters and they are identical, right? There has not been any kind of crossing over yet. Like this is just a sister chromatid. When we talk about the homolog, right? So remember, we are diploid organisms. So this is, let's say, chromosome 1, one came from mom, one came from dad. So these are homologs, which means they have, your similar genes, right? So like, let's say this is the gene for dimples, gene for dimples. This is the gene for widow's peak, gene for widow's peak. So they're going to have the same genes at the same loci, but your allele variation is going to be different. And so that's why the homolog is not the twin, because those, mom and dad give you a huge variety of genes. We know that. All right, so I appreciate your questions. I appreciate all of you who are interacting with Jed and anyone else who's helping out. I really do. I love your questions, so keep them coming. Thank you. Okay. Next question. If a cell has 16 centromeres, how many chromatids does it have? Good. Yeah. So the centromere is the thing that holds your chromatids together. So a lot of you got that right. Really good job. Again, this would be a question where I would just draw it, right? So draw out what you've got. If there are 16 centromeres, think of it like a button in the middle, right? How many chromatids, individual chromatids would you have? And that would be 32. This doesn't like me going next. Okay. Oh, here we go. So if a tetraploid cell undergoes meiosis, what's the ploidy of the daughter cell? So a lot of you answered. So it would be 2N, right? So if we are originally a 4N cell, I would want my daughter cells to be half of that after meiosis. So you'd want it to be a 2N cell. I don't even know if anything exists like that. I was just trying to think of an application question where it's something you've never seen before, but it's following that same guidelines of like, your ploidy needs to be cut in half with meiosis. Okay, this image best illustrates. Good. Independent assortment. You all got it for the most part. Great job. It showed independent assortment. Okay, so now we're on to just a couple random evolution questions I threw in there. So phenotypic plasticity absolutely has to do with if the environment changes and affects the phenotype, then it's phenotypically plastic. It can be changed. On Mentimeter. Okay. To determine an unknown genotype, you prefer a test cross. So you're going to, good job. So 300 of you answered that correctly. Really, really nice. If you don't know a genotype, you can cross it with something that you know for sure based on the phenotype what the genotype is. So if we're talking about just simple, Mendelian genetics, recessive, if I look at something and I know that it has a recessive trait, like phenotypically I know like, oh, that white bunny, I know white is recessive. And so if that bunny is white, it's going to be homozygous recessive. So I want to test cross with a known genotype to figure out what the other one is, right? So based on like, hey, if it's homozygous dominant, what would the outcome be? And if it's heterozygous crossed with homozygous recessive, what would it be? We don't have too many left. Come on, next. During DNA replication, good, most of you got that right, the leading strand. We just talked about it, so excellent job. Is that the last one? DNA replication, excellent. Good review. Most of you got that right. It's happening during the S phase. Man. When we say a gene is expressed, it means, yay! All of you found my trick question. So yes, all of these are true. So the gene is not inhibited. Protein synthesis is happening. Transcription and translation are happening when a gene is expressed. Excellent job. How do you remember the difference? Okay, so this was just for us to kind of look at together. I wanted you all to type in, how do you remember the difference between an inducible operon and a repressible? So someone said inducible is turned on and repressible is turned off. Inducible means it will need to be turned on by an enzyme binding to the promoter. Repressible is turned on until an enzyme binds and inhibits it. Good. I'm trying to see if there's other ones that would be good. I think they're all really good, by the way. Sorry, I'm just like learning. I'm looking at these things. So it's okay if you don't remember. I see some people being like, I don't know. I've never learned it. And like, remember, that is okay. That is okay. This course has so much to it. Okay. If you didn't learn one tiny thing, it's not going to make or break your score. I promise. Okay. But I will go over those operons in just a moment. Eukaryotic cells regulate transcription at the chromatin level, by the real answer would be methylation and acetylation. So when I say the chromatin level, I didn't say the nucleotide level, right? So chromatin is the structure of DNA when it's kind of all jumbled up and it's got I guess I could have written the word histone level. That might have been a better question, but it's through methylation and acetylation. And I do have a slide on that, so we will talk about that, okay? What confuses you most about evolution? Okay, 4 laws, weird tree diagrams, yada, 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 trees. Okay, good. Probability, Hardy-Weinberg. All right, we'll talk about all that. We're doing it. We're going for this, guys. We're going to get it done. I just was curious about what you thought of with the natural selection. I see lots of Darwin, lots of fitness, which I like. All right, so last question, I believe it's Hardy-Weinberg. If we have 40% recessive, calculate the frequency of the dominant allele. The last answer option is your correct answer, just because I wanted to show you that frequencies can be shown in percentages and decimals. It's the same number though, right? 37% is 0.37. So just kind of a quick math thing, just to make sure you know that if you're given a percentage, how to get it into an actual number that you can throw into a Hardy-Weinberg problem. For those of you who did not know how to do this one, because I see a lot of you answered 60%, and that's probably because you use the equation P plus Q equals 1. However, here this problem is giving you the recessive phenotype, okay? And I know this is jumping ahead a bit, but I think it's important. Well, I just said it again. Oh my gosh, you guys, everyone, help me. I live in Michigan, so it's almost 9 o'clock, which I know to you is not that late, but I am a mom with 3 kids, so almost 9 o'clock is like midnight for me. So if I say something and you're like, what is she talking about? Then, I'm just so sorry. So anyway, I want to go over this Hardy-Weinberg one real quick. So Hardy-Weinberg is an equation we use to see if a population is evolving, if it's changing, okay? If we say it's in Hardy-Weinberg equilibrium, that means you can use these equations. And how we would use it to see if it's evolving, you basically do the equation, figure out what your allele frequencies or genotypic frequencies are, and then you would do it again, maybe the next year or 10 years later. And you'd be able to see like, hey, whoa, look at, the recessive allele was 80%. Now it's 20. What's going on? The population's evolving. So in the problem I gave you, I said that the dominant phenotype was 40%. So that, there's 2 key words here. Phenotype for dominance, we know can be 2 different genotypes, right? So I'm going to use the letter H. Okay, so it could be homozygous dominant or it could be heterozygous. So if you are given a dominant phenotype, or they might not even say phenotype, they might just say like, oh, the amount of, brown bunnies are dominant to white, and there are 750 brown bunnies. They have not given you any of these letters yet, right? They haven't. They've given you HA, or, dominant, homozygous dominant and heterozygous. So you can't really do anything with that number yet in terms of this equation. But what you can do is go, hey, well, if the dominant phenotype is 40, well, then the recessive genotype has to be 60. And that's just not even using the equation. That's just using like common sense of like, well, if 40% are showing the dominant phenotype, then 60% are showing the recessive phenotype. And remember, if we're talking about a recessive gene, genotype is phenotype here, right? So I would have 60% being recessive. Now, I cannot use this equation yet. The equation sheet that you'll be given on the test will tell you that that these are alleles, right? So individual allele. So unless you see like, oh, what is, the allele is whatever, you can't use P plus Q yet. And so I think that's what a lot of you did right from the beginning. You just did, oh, if this is 40, this is 60, there's my allele. So let me just go back to the question to make sure I know what I was asking you guys in the first place. Calculate the dominant allele. Okay, so the question was wanting to know what's the dominant allele. So we were hoping for you to solve for P. That's my question mark. The dominant allele. Allele is not plural in this case, so P isn't plural. P squared is plural. Right? So if we go back to like what these all mean, P squared is your dominant. 2PQ is your heterozygous. Q squared is your homozygous recessive, right? Because it's little, Q times Q, H times H. So anyway, we get our recessive. We know it's 60%, which we can change to 0.60 pretty easily. So this is what we've been given, right? So you need to take, we know that Q squared equals 0.6. So you take the square root, okay, right? So just remember math. How do you get rid of a square? You square root it. And unfortunately, I should have chosen a better number. Hold on a second. I might have even done my math wrong here, everyone. I'm losing my mind. I think I might have. P is 0.37. Yeah, I did do my math wrong. Ugh, so sorry. Goodness gracious. I think I solved the problem thinking that the dominant phenotype was 60%. That's what I did. So, okay. Hold up. We'll just keep going with this math, okay? So if you take the square root of 0.6, I'm telling you, I was multitasking today and I shouldn't have done it. I just flipped my things. So if you take the square root of 0.6, you get 0.77. So Q is 0.77, which means P is 1 minus 0.77. So you get 0.23. And then it's asking for what your dominant allele is. So that would be the answer, 0.23. So goodness, my bad for flipping that up. I think I thought I put, oh wait, no. Oh my gosh, you guys, look at me. I'm losing my mind. The question did say recessive phenotypes. Ay. I'm so sorry. You guys are probably like talking in the chat. How terrible right now. I'm losing it. But still, what I showed you here is legitimate, that if they're giving you a phenotype, this is what you have to do. Okay? So this is exactly what you have to do. In that practice problem, I was too easy on you, and I gave you the recessive phenotype was 40%. Okay, so the recessive phenotype being 40%, so that you should know, okay, it's given us my Q squared of 0.4. I need to square root. You square root, and that's where you get your 0.63, and then your P becomes 1 minus that, which is 0.37. Okay, sorry about that. I was so excited to teach you the one trick that like I know people get mixed up on, and so I screwed it up. So my bad. So sorry. It's one of those nights already, I'm telling you. Okay, let's move it on. So how are genes regulated, right? I was talking about the chromatin level earlier, and I mentioned how at the chromatin level, or I should have said histone level, you can add methyl groups or acetyl groups to make the DNA less or more accessible. Methyl is a carbon with 3 hydrogens, so it is nonpolar and it doesn't like water. So it is going to clump all these things together and it's going to hide the gene. Acetyl groups like water, okay? They're polar, they dig it, they open up the gene. So these are simple ways that epigenetically we can, or not we, it's not like I'm in charge of this, but that eukarya can regulate their gene expression, okay? It's epigenetics because it's epi on the outside, like your skin is the epidermis. So on the outside of your skin, or the top of your body. So this is on the outside of the DNA. It's not changing the nucleotide sequences at all. Another way eukaryotic cells regulate are by both sequences and proteins. Okay, so be careful when you see language like that. And if you're getting a model or a picture or a diagram and they're talking about either sequences or proteins, pay attention to that. Those words matter. If it's a sequence, it's a part of the DNA itself. It might not be the gene, but like this promoter, it's a sequence. It's an actual sequence of nucleotides, right? So TA, right? The Tata box. That's a sequence. You have enhancers, right? Enhancing sequences. But you also have proteins like a DNA binding protein or transcription factors, activators. These are all proteins that also help RNA polymerase do its thing, which is, remember, if we're expressing genes, we're transcribing them. Okay, so this is regulation of gene expression. So we're regulating, is it easy for RNA polymerase to come in and transcribe, or is it not? Think about why you'd want to do that. There are certain times maybe the environment changes and hey, you know what? This organism needs to be able to do more of something, right? Maybe it needs more testosterone. Maybe it needs less of something. So this ability, this kind of ties back into your cell communication and how there's so many different ways to do it because there's so many different things that need communicating. Regulation of gene expression is the exact same thing, okay? Where there's a huge variety of genes, huge variety of either needs to turn on or turn off. In prokaryote, remember prokaryotic cells don't have a nucleus, so they also don't have as much DNA, right? They've got a circular piece of DNA, not the plasmid, I'm talking about the other part. But an operon is an example of a way prokaryotic genes can regulate, which means turn on or turn off their gene expression, okay? I like to use the analogy that if it's a sunny day, it's the brightest day, and you happen to live somewhere that has lots of windows, it would be a huge waste of energy to have every single light on, right? Because you've got natural light. Why would you turn the light on? It's a huge waste of energy, okay? Operons are the same way. Look at this picture here. So we have this operon where it can make a bunch of these enzymes. What do these enzymes do? Well, they help digest lactose. But if there's no lactose in this environment for this prokaryotic cell, why would it waste the energy making all this stuff, right? It wouldn't, and it shouldn't, okay? Energy is key, and I'll get into that in ecology in a little bit, but when you think of living things, being able to take in energy, like, think of cellular respiration and photosynthesis. We don't want to waste anything. We don't want to waste energy. It's a lot of work to be fed. It's a lot of work to stay alive, and it's a lot of work to reproduce. Okay, so if there's a way to not waste energy and not transcribe and translate something you don't need, then that's awesome. And that ties into evolution as well, right? So this is something that has evolved over time, and it makes this organism fit that is able to do this. So this is, we looked at the word inducible or repressible. So looking at this one right here, it's showing that you've got this upstream gene that is turned on and it makes this repressor protein. This repressor protein, and when it's made, it is already active. It is an active repressor protein. So that means when this repressor protein is made, this operon, which is all the time, by the way, this operon is on the operator. This repressor is on the operator. So RNA polymerase cannot transcribe. When the environment changes and lactose is present, it is a co-repressor, and it's not technically lactose. It's like beta something. But either way, lactose is in the environment. It binds to the repressor, and the repressor changes shape. So it pops off, and RNA polymerase can go ahead and transcribe. Eventually, we get a little bit of negative feedback, right? So once lactose has been digested by all this stuff, we don't have any more, and it can go back to repressing. So this is an example of an inducible operon. It can be turned on. It can be turned on. It's normally off, but it can be induced. Repressible is the opposite. And I don't have a picture of that because we have more to do, but a repressible operon can be turned off, okay? I have an example of a free response question that has to do with gene expression, okay? I know we only have an hour left, and so I'm going to try my best to, let's see. That's all I have for this unit, so I'm going to let you do it. I'm going to let you look at this free response, okay? I'm going to give you a second to read it, and then I'm going to slide over to what kind of questions they're asking you, okay? Okay, so even if you're still reading, I'm just going to start talking. When you get a diagram like this, I would recommend taking a look at it and just try to annotate it yourself first, right? Unless there's something that you have no idea and you're like, I have no idea, right? But this is a gene, right? So annotate, like this is DNA right here. Then it's moving to this thing called TDF. If you did read first, you could go up and be like, okay, well, what's TDF? Protein. So I would write the word protein on top of this box. Or if you're not taking this on paper, I would just quickly on a piece of paper write out the word SRY gene, arrow TDF, and I'd write the word protein right there. Okay, just to help make sense of what all these boxes are, because it's really easy to mix up and label something a gene when it's not, or label something a protein when it's not. Okay, and a lot of the information they will give you. So it tells you, okay, this SRY gene codes for a protein. Here's my protein, TDF. Okay, it's a transcription factor for other downstream genes on the Y chromosome, like the SOX9 gene. Here's another gene. So I'd label gene. Okay, so gene, protein, gene. TDF also induces differentiation of these cells, which also help with development. Okay, so there you go. This table gives you how many base pairs are in the transcribed region and then how many are in the translated region. Okay, it tells you there's 1 exon and 0 introns. Okay, that information is actually really important. So the first question we're just going to do together, it says circle the arrow that most likely represents protein synthesis. Okay, so I'm going to go back. Which arrow best represents protein synthesis specifically? So hopefully you thought of this first arrow, right? Because we're going from gene to protein. That's protein synthesis right there. I've had a few students who have showed this question to before, and they're so used to thinking of like, they need to look for 2 arrows because like first this needs to be transcription, the other one needs to be translation. But this is just a model, right? It's a model that's kind of showing, hey, here's the original gene, boom, there's the protein. It doesn't need to show you, oh, here's the mRNA and here's the mRNA processing. It's just a model. So read the diagram, label and annotate, and go with what you know. B says, according to the table, to the information in table 1, calculate the number of amino acids present in the primary amino acid sequence of the final SRY protein, otherwise known as TDF. So it wants to know, calculate the number of amino acids. Okay, how many amino acids would be there? So, and it's wanting to know number of amino acids. Okay, so if you think of amino acids, your mind should go to translation, right? So the translated region of the gene, right, which is going to be your mRNA, has 612 base pairs. So if I have 612 base pairs, that should make you think, okay, what's that related to amino acids? How would I figure out the number of amino acids? And hopefully the ratio of 3 to 1 comes to your head. That for every 3 base pairs, there's 1 amino acid. So 612 divided by 3 is 204. So it'd be 204 amino acids that build this protein. Okay. Another question, if SRY has no introns, well then why are the translated regions different than the transcribed regions? This is a really hard question. Again, this is kind of an out there question. I wrote it, but I just wanted to show you that sometimes there's a question you might not know the answer to, and it might take you forever, and it might be best just to move on, right? Go to the ones you know. But what I want you to think about is, if I have a transcribed region, right, remember what happens in eukaryotic cells, how you get like this addition of this huge poly A tail, right? And a huge region for that poly A tail is that as soon as that mRNA leaves the nucleus, those poly A tails start getting eaten up. Okay, and so that's what this is representing here. It's a very specific thing that I wanted to bring up. Like I said, the chances of them asking you that kind of question, but still zooming out and being like, well, why would that ever be bigger? What happens in between leaving the nucleus and coming to the ribosome is that you lose base pairs. Obviously not important ones, but still you lose them. Okay, these are predict questions. Let me see if this one is good. I'm going to skip those actually because we're running out of time. If we've got time, we'll go back. But when you get to predict questions, don't be afraid to make a prediction. Usually with predict questions, there's a variety of answers that could be true. Okay, so don't think that they're expecting you to, read their minds, but just use your science, use your biology knowledge. You know more than you think you do. Okay, be confident. You've put in the work. You know the stuff. And if you have to, remember, just write down stuff you know. Write down the stuff you know. Draw a picture of something that you know to be true and make your best prediction. Okay, so evolution, we're moving on. We're doing good. We're going to have about a half hour for each. So big themes in evolution. We've got natural selection, population genetics, Hardy-Weinberg, evidence of evolution, phylogeny, and then speciation and extinction. So here we go. Microevolution has the word micro in it, so it's a change in allele frequencies. Remember, alleles are that variation of the gene. So when I talked earlier about that original population to the bottleneck population, that's a huge change in allele frequencies, right? We used to have a variety of alleles, so not only variation was changed, but allele frequencies were changed. Other things, I didn't want to start just with natural selection because I feel like everyone starts with natural selection. Ooh, sorry, I just got interrupted by a question, and you are all so brilliant, and I love it. You guys asked a question about if a stop codon counts as an amino acid, and it wouldn't, but on the test, they're really just looking at your knowledge of the 3-to-1 ratio. So I don't think they would expect you to subtract one amino acid, but that's an awesome revelation from you all. So just to kind of recap, when we divided 612 by 3 and you got 204, a lot of you realized, hey, we've got to start codon. And here's the other thing too. Methionine, you know how that's the very first amino acid? That's usually like a non-functional amino acid that gets cleaved off anyway. So, and that's something that I don't think they expect you to know either. So again, you could answer it as 204 or 203, and I'm sure they'd still give it to you because there's a lot more to all of this than we have to worry about in terms of this course, okay? So really that kind of question is just looking at, do you know the 3-to-1 ratio of base to amino acid? But really great job, everybody, with participating and asking good questions. Okay. So microevolution is change in allele frequencies. It doesn't just happen by natural selection. It can happen by mutations, gene flow, which is, the flow of genes in and out, emigration, immigration. Genetic drift, so your bottleneck effect, founder effect, and non-random mating. So that's having to do with sexual selection. So the idea of like, hey, this female peacock isn't just randomly picking one. She's choosing the one that has the more eye spots. That is specific. It is not random. And so that'll change the allele frequencies, right? You see less peacocks with a small number of eyes because the allele frequency has changed over time so that the males have tons of these beautiful eyes on the feathers. So if we think about a story of evolution in terms of natural selection, so I'm zooming in on natural selection now. It has the word nature in it and has the word select. So nature is selecting a specific phenotype, right? The classic moth on the tree in the Industrial Revolution example. Moths are eaten by birds. Birds are visual predators. And so if the moths are camouflaging, then the birds are going to not eat them as much, right? So if you were to tell the story of natural selection about the moths, you want to first talk about you want to make sure you don't talk about individuals adapting or individuals changing. That cannot happen. Okay, that cannot happen at all. So what I would say is, in this existing population, there was variation, right? So the existing variation was that there was a variety of moth colors. When the environment changed, right, or even if the environment doesn't change, there's always going to be a struggle to survive, right? And there's going to be certain phenotypes that do better. Your teacher might have called it overproduction or over-reproduction. Or talked about competition. But either way, there's a struggle to survive, right? Maybe because of shelter, maybe because of access to water, maybe because of access to whatever. But in this moth example, the struggle to survive is because of the color, right? And so when everything got dark from pollution, the darker moths fared better. We would say their evolutionary fitness was better. They were more likely to survive because they didn't stand out. And if you are surviving, guess what? You get to reproduce, right? So that's like the big thing of natural selection is the existing variation or existing phenotype variation has better fitness. And over time, that is the evolutionary fitness allows them to survive and reproduce. And we see larger or a bit, increase in allele frequencies of that typical variation. It can change drastically back and forth. Hopefully your teacher talked about the, I think I have it in here. Hold on. Did not keep here. So this example of your ground finches on the island of Daphne Major. So these two scientists were there and gathered every single finch on the island, measured their beak. No joke. Every single one. And they did this for like 20 years. It's amazing. And they got to go to the Galapagos every summer. Why? I'm like, why? How do I get that job? So anyway, they had this original parent population with these beak depths, right? How deep their beaks were, goodness. And the number of birds who had that, right? So in white, this is the parent population. The black bars are going to be the survivors of the drought. So there was a drought in 1976. And as you can see, those with deeper beaks survived. They fared better, right? They were more likely to survive. But this doesn't tell the whole story. With natural selection, we have to also then look at their offspring. So we know who survived was the 9 and 10s, right? The beak depth here. Look how little, 7.3 beak depth survived. They only found like 1 or 2 birds. And so check it out. The original population has this kind of a curve, and this is what their offspring looks like. It's shot to the right a little bit. And that's because of the drought. And so they continue taking data and looking at this. When the drought reversed, guess what happened? We went directional selection that way. Okay. So again, this is something that comes up later too, is directional selection. It's this idea of the allele frequencies are changing and selection is happening in one direction, right? So either one favorable side to the right or to the left. You might talk about like heterozygous advantage. So that would mean like those with the heterozygous trait are doing well. And so your good example there is sickle cell anemia, right? Because the heterozygote, even though, they're carrying that recessive allele, and you would think the recessive allele would decrease like crazy, it doesn't, because it actually provides immunity to malaria. And so that is true in certain populations of Africa. Okay, so just real quick, real summary of natural selection. There needs to be variation that already exists. There's over reproduction. So basically that just means not everyone can survive, right? It's the harsh truth that not everybody can survive. And then there's some sort of selection which comes from, predation, resources, climate change, or just climate in general, right? And then those who survive pass those genes on. Okay, so make sure you are able to talk about all 4 of those things. We did do a little bit of Hardy-Weinberg already, but so just as a quick review, we are able to figure out whether or not an organism or a population is changing, right? Is it changing? So we have our P, which is your frequency of allele 1. I usually call it the dominant. And Q is frequency of allele 2, allele singular, Q singular. When you square those, Q squared, that means I have 2 of the same allele. So I'm a homozygous genotype. P, if I square that, that's another homozygous genotype. And 2PQ has 1 of each allele, so that's your heterozygous. Okay, so let's try a practice problem. If the frequency of the recessive allele is 0.6, so be careful. The recessive allele is 0.6. Determine those 3 things. And I promise I won't mess up the math this time. Oh, man. All right, so when we have our recessive allele of 0.6, make sure you're looking at just the singular Q. So that means we've given you Q. So you can pretty easily solve for P, right? So P is going to be 0.4. Okay, so that's your dominant allele frequency is 0.4. Then your frequency of the heterozygote should be pretty easy as well, because then you're just going to do 2 times P times Q, right? So 2 times 0.6 times 0.4. So that should be 0.48. So frequency of the heterozygote is 0.48. And then here's the only one that might have thrown you for a loop, is the frequency of the dominant phenotype. Okay, so you would have had to first solve for your dominant, homozygous dominant genotype. We already have our 2PQ. So P squared is going to be 0.4 times 0.4. So 0.16. Okay, so you have to add now 0.16 to 0.48. Okay, so 0.64, 64% is the frequency of your dominant phenotype. So again, just be careful with your Hardy-Weinberg. If they've given you a phenotype, be like, hey, if it's the dominant phenotype, that's talking about P squared and 2PQ. So don't try to do any wild math problems where you're like trying to solve with a quadratic or something. You're right. Start with this equation. Okay, start with this equation if you can, unless they've given you this Q squared, right? So if they've given you the recessive phenotype, then by all means, start with Q squared and get it to Q. All right. So moving on to the deeper part of our evolution unit, and we will get to a tree. So we think about evolution, it's not just about things changing. It's about how all things are unified together, right? How all living things are, well, we're all alive, first of all. We're all made up of cells, right? And we also have very specific things in common as well. We have ribosomes, some sort of genetic material, and an outer membrane, something that separates our outside environment, external environment to the internal. So if I simplified the tree of all living things, right? We have bacteria, archaea, and eukarya. All right. Making sure you know these, I think, would be nice, knowing that they all have ribosomes in DNA. Okay. Cellular structures that unify all eukaryotic cells. So we're kind of zooming in on that big tree of life here to show where what other things are in eukarya. So that's where your animals, fungi, plants, all that. The things we all have that prokaryotic cells do not, we have membrane-bound organelles, we have linear chromosomes, and we have genes with introns. And again, I know I'm tying in old units now, but that's kind of the whole point, right? So we learned about prokarya versus eukarya in unit 1 or 2, right? You learned about linear chromosomes versus circular in some other unit. We learned about introns in the gene expression unit. So be okay when you see things that are cross-linking, right? There will be problems where that happens, and rest in your knowledge and be okay with it, because you know a lot more than you think. So again, these are all things they have in common compared to your prokaryotic cells. And this helps unify living things. Another thing that we want to talk about with evolution is, how do we know things are changing, right? What are these things? Biggest one, as far as how rock solid the evidence is biochemical, right? So like if you're at a crime scene and there's been some, like a hand print or a shoe print left, that's kind of like fossil evidence. It's not that great. It's okay, right? Especially if it's a hand print with like no fingerprints. It's okay. It's an impression. We can make some hypotheses about it and try to find the bad guy or girl, whatever. But with biochemical, that would be like, hey, they left DNA at the scene. They left hair or blood or whatever, right? We can actually sequence who this person is. We can use gel electrophoresis on that. And feel free to reach out to Jed if you guys want me to talk about gel electrophoresis, because I think we will have some time. Because I kind of skipped over that and just did DNA cloning. Anyway, so biochemical evidence is our number one evidence in terms of rock solid. It really helps us see who is more similar, which organisms are more related, who is the common ancestor, who's the most recent common ancestor. And remember, that's because, hey, we all have DNA, and this is the ultimate code, the ultimate information that makes every organism who they are, right? In terms of physical traits and how the body works and organisms or cells work. Morphological structures, when you think of morphology, that's physical structures, okay? There's homologous, vestigial, and analogous. Homologous is going to indicate common ancestry. So here's a picture of homologous structures. We have, even though they look different, right? They kind of look different. The whale, the bird, the dog, us. But look at the body plan. One of my favorite scientists, what's his name? Now I'm not thinking it is Phil Shubin. He's a professor at the University of Chicago. He's in a lot of those videos, like Your Inner Fish, if you've seen it. I'm sure your teacher has shown it to you. Anyways, he says something in that movie where he says they follow the same body plan. He goes, there's one bone, two bone, many bones, digits. Look at the dog. One bone, two bone, many bones, digits, so on and so forth, right? So same body plan. Where did that body plan come from? Boop, DNA, right? So this indicates these are homologous structures. They might have different functions, but they're homologous in terms of the structure. Analogous structures are different, right? They have similar functions. They might like help an animal fly or swim, but they don't have a similar structure. All right. Is evolution ongoing? Absolutely. Right? We're seeing this right now with the coronavirus, right? So it is changing. We're talking about different strains, right? And so evolution is absolutely ongoing. This example is just talking about, this is from the CDC. This is talking about just how bacteria have changed and how their certain strains or certain bacteria are surviving and they're able to become antibiotic resistant. So don't be surprised if you see some question about antibiotic resistance, because that could kind of tie in some gene technology. It could also tie in evolution and talking about how if you only need one bacteria to be resistant, and it's not resistant because it is better than the other ones. It just probably has a mutation that for whatever reason makes its cell wall impermeable to the antibiotic, if that's how the antibiotic is going after it, right? If it's trying to get into the cell wall so it bursts. But yeah, it's one little mutation that could happen, right? And give you that variation. Now, when we talk about macroevolution, this is where we're talking about speciation, right? So macro meaning big. We're not looking at population genetics and how, we have, 20% green and 80% yellow. We're talking about big picture, macroevolution over a long, long time. And then we get these speciation events, right? Where we get divergence, where at each of these nodes, we get a splitting, and then eventually a bunch of different species. Real quick to define a species, it's the most commonly accepted. You probably learned about like the 4 different types, but really what you need to know is, hey, they can interbreed, their offspring are viable and fertile, which means they can live and they can have their own babies that can also have their own babies. So they're viable and fertile. And they don't interbreed with other populations. How this happens, how a species happens, is that gene flow has to stop. So if you're an organism, if you're like a population of zebras and you somehow as a group get separated geographically, for example, and the gene flow between those two groups stops, you become isolated. Eventually, that geographic isolation could lead to reproductive isolation, which means you aren't able to interbreed with each other. And we would call you two new species. Okay? If halfway through this speciation event, gene flow starts to happen because maybe a couple zebras come over and they mate with another one, and then they're still genetically close enough to produce viable, fertile offspring, well, then speciation is not going to happen, right? So gene flow has to completely stop. So in this example of fruit flies, we like to see, fruit flies are great because they've got a lot of traits that we can physically see. We can be like, eyeball color, wing shape, body shape, boom. Very easy to work with, and they reproduce very easily. So if these two initial sample were separated and put on completely different food sources, so these were put on a starch and these were put on maltose, and then after many generations, all of a sudden there's a mating preference, and they're like, hey, look I actually prefer this and these prefer that. We're not saying that changed because they wanted it to. There was some existing, that population changed over that time. They were using a different food source. Over time, just naturally, remember, variation happens, mutation happens. Variation happens because, remember, meiosis, right? So Other ways to prevent gene flow, like I said, speciation can happen geographically. So we can have like separation here. So this would be allopatric speciation. They're separated, A for away, A for away. It's kind of like allosteric inhibition of your enzymes. And then you can have sympatric, where they're like close by, but they still speciate. So here would be like you have temporal, so you're not awake at the same time, right? So maybe for whatever reason, your portion of the population, isn't awake when the other one is. And over time, you can become sympatrically speciated. Or behaviorally, like, hey, I don't recognize that song. And then over time, hey, look we've got a whole new species. Or polyploidy, right? So this one became 28, or these two were brought together, and all of a sudden we have your diploid number of 42. And we get this new species that then can either self-pollinate. Usually it happens with polyploidy. They can self-pollinate, and we get a whole new species there. I wouldn't worry about being able to name each of these. I think you're going to be asked to apply this kind of stuff. Okay, I'm going to move on. So cladogram versus phylogenetic tree. It is on the CED that you know the difference, but honestly, I think every teacher kind of teaches it the same, but I'm going to say it just because it was in the CED. Phylogenetic trees estimate the timing. So you might have like little numbers that show you like, oh, 100 million years ago this happened, or 200 million years ago this happened. So phylogenetic, just think, has the word T in it for time. So there's a time component. That doesn't mean every phylogenetic tree has to show time necessarily, okay? But notice how like all these animals are kind of, like they're not all vertically aligned, where with a cladogram, they are all aligned because there is no time issue. We're just looking at evolutionary relationships. At a very basic level of understanding trees, the base is our common ancestor. And so whatever traits are here, whatever characteristics are here, everything above it has it. These two trees are identical. You can see them in these boxy ways or these straight ways, either one, whatever is your preference. The outgroup is going to be the group that is least similar to everyone else, right? Think of like, oh, the outcast. I know that's bad to say, but my son's in first grade, and so he's kind of learning that, like, importance of including people and don't let anyone feel like they're on the outside. So anyway, C is going to be your outgroup. They are least similar to the other two, right? C is your outgroup. So when you're building a phylogenetic tree, I always like to build the outgroup first. And so those are your nodes, like I said. All right. So how do you make a tree? First, they used to use physical comparisons, so morphology or phenotypes. Fossil evidence can still be used today, absolutely, but the best data comes from DNA and amino acids, right? The best data comes from DNA and amino acids. It does. You may be given a tree that's already done, and you have to analyze it. So for example, which organisms contain vascular tissue? So you go, okay, where's vascular tissue? Where is it? Where is it? Here it is, vascular tissue. So this characteristic came about after the mosses and hornworts, but before all of these other ones. So this is a clade right here. So the moss, the club mosses, the ferns, the gymnosperms, and flowering plants, everybody has that vascular tissue. Okay. You might also be asked something like, oh, who are the descendants of the club mosses? Who descended? Think of that like Disney movie. It's the kids of the evil people from Disney. So the descendants of the club mosses are everyone above that, the ferns, the gymnosperms, the flowering plants. Okay, so I think that'd be a good analysis question. Okay, if we were to build this tree, I'm going to give you a few minutes to take a look. It tells you the table above shows the percent differences of amino acids for organisms 1 through 4. So organisms 1 through 4. Notice that these blocks are blank, and that's okay because you don't need to compare everything twice, right? So like if I'm comparing organism 1 and organism 2, and they have 15% difference, I don't need to re-compare them right here. I mean, I could, but then the table would be a huge mess and you would all be really mad. So take a moment, think about what you're seeing, draw, and figure out which ones would go where. Ooh, okay. So I'm going to hop in. I know you're probably still thinking. I like to identify the outgroup first. So try to figure out which organism seems most different from all the other ones, right? So check it out. We've got organism 3, who's got 30 differences with organism 1. It's got 28 differences with organism 2, and it has 29 differences with organism 4. So looking at that, I would jump in and say organism 3 has to be right there. That's where I like to start. I like to identify the outgroup first. Then I actually like to find the 2 that are most similar. And not every tree will look like this. Some are going to look, you might have something like that where you have 5 organisms, right? But the same idea holds still, that like these 2 are more similar to each other and these 2 are more similar to each other. Oh, and here's the outgroup, sorry. Anyways, so the 2 that are next most similar, I would look at the smallest number because we're looking at differences, okay? So the least amount of differences means the most similarity. So organism 1 and 4 are going to be here. It doesn't matter where you put 1 and 4 because this node can swivel, right? So it could be either 1 and 4 or 4 and 1, you'd get the same amount of points. And then process elimination, or you could look at the numbers, 2 would be there. Okay, this is a pretty simple phylogenetic tree, but I thought just for the purposes of review, I would do something simple so that you all felt pretty good about it and you could just rest on the basics, because if you get something more difficult, just remember your basics and just problem solve. Take your time. Okay, providing reasoning for the placements. When they ask you to reason or justify, give numbers. Okay, so say, 1 and 4 have only 7% differences, so they are more alike. Organism 3 has 30, 28, and 29. It is most different, so it is in the outgroup. Okay, and it's okay if you then say, and then 2 is placed there because process elimination, it's kind of in the middle. Circling all the descendants from organism 2. So I mentioned that before. Your descendants from organism 2 are going to be everybody after organism 2. Okay. Your friend claims that DNA sequences will provide more accurate information to determine your relationships between organisms compared to amino acid. Okay, so these are amino acid sequences, and your friend's saying, well, actually, it's DNA that's better. And you're like, oh gosh, okay. Well, let's stick with it. Provide one piece of reasoning to support their claim. So how could you support them? Why could DNA be better? So one thing that hopefully starts to come to mind is that, well, amino acids come from DNA, right? And amino acids, let's say I have these 3 amino acids, right? Let's say I have lysine, I have arginine, and I have guanine. And by the way, you do not need to know those names. I just know them because I've been doing this forever. So I have these 3 amino acids. The codons that can code for these, there's like 4 different codons for each of these, right? Remember that chart, how that last base, that third base could be like 4 options for a lot of them? So because of the redundancy of the code, one amino acid could have 4 different codons. So ultimately, DNA is the original source of information, so it's going to be more accurate. Okay, so if you said something like that, or the each amino acid can be coded by a variety of each amino acid can be coded by a variety of codons, and so that helps. Last one. Prior to gene technology, where scientists can compare DNA and RNA sequences and the primary transcript of proteins, they relied on just observing morphological evidence to infer relationships. So describe 2 types of morphological evidence and explain whether or not they can indicate common ancestry. I included this one because this is another great example of questions where there's more than 2 types of morphological evidence. They're just asking for 2. So in a way, like if you don't know everything, that's okay, because they might not ask for everything. They might ask for 1 or 2 aspects of something, and you might just know 1 or 2 things. You know what I'm saying? So here I'd be looking for an answer that talks about homologous and analogous structures and then explaining whether or not they indicate common ancestry. So explain isn't just, oh, it does not indicate common ancestry. Explain it, right? Why? Not just what. Describe is more like what. Explains like why. Tell me why. Okay, why? All right, here we go. Let's move on. Let me delete this. Moving on to ecology, people. We are done with evolution. And like I said, this is a cram. We are crammed. Our brains are crammed. Even the teacher's brain is crammed. Okay, I can do this. 25 minutes. Ecology. We're going to go over animal behavior, carrying capacity, energy flow, and human impact. Oh, and the biodiversity equation. So we think about animal behavior. Animals respond to changes in their environment. Be aware of those words behaviorally and physiologically. Behavior, it's stuff that we can see, like, oh, look, it's leaning towards the sun. Or, oh, look, these guys are yelling out to establish dominance. Or this guy's doing a dance to try to find a mate. That's behavioral. Physiological would be how they respond internally. Like, is their metabolism increasing because they're cold? Or is their metabolism decreasing because they're hot? Okay. Those are things you should know. Physiologically would be inside. Another thing about behavior is that whatever the behavior you're talking about, if it's a behavior that is present, it's probably increasing their fitness. If it wasn't increasing their fitness, it probably would not be a behavior that lasts, right? So if there was an organism that behaved in a way where like it kicked its chicks out of the nest way too early, that behavior probably wouldn't continue in generations because those chicks are now gone, right? Because it was too early. So its genes, whatever genetic component that made that bird do that, is going to die with that bird, right? Because it didn't increase the fitness because now those chicks are gone. I'm sorry, that was a really bad, sad example. I don't know why I used that. Sorry. Oh my goodness. It's almost 10 o'clock here. So I start to use like more yikes examples, but maybe it'll be something you remember. Anyway, organisms, they communicate visually, audibly, tactically, electrically, chemically. So make sure you know those words too. Tactile means like touch. So it'd be like, animals when they're like grooming each other, think of like chimpanzees, that's tactile communication. Visual is what we see, audible is what we hear. Electrical would be like organisms in the water that can use those sorry, bad example, but you get the idea of what electric is. And chemical would be like pheromones, right? Like releasing certain smells. And yeah, okay. Energy flow. Be aware of the words endotherm and ectotherm. So if you're an endotherm, we maintain a pretty stable body temperature, okay? Because our metabolism can regulate it. If we're cold, it can increase metabolism to help heat us up. If we're freezing or if we're warm, it can decrease to kind of cool us off. And obviously we have behavioral ways to also regulate our body temperature. But if we're just talking about ectotherm versus endotherm and metabolism, that's why I'm just sticking to that. Where with a fish, right, its body temperature changes with the day, right? So water is pretty cold in the morning. Sun comes out, warms it up. The fish feels warmer. It doesn't have a way to regulate its body temperature in the water except for behavior. So with ectotherms, they do things behaviorally. So like sit on a rock to warm up, or they move to shade, or maybe they dig themselves under the sand to cool off. I talked about this earlier, but again, this is a huge, big picture thing that it's all about the energy. So if you're reading something, think of it in terms of energy. And this is similar to the disruption thing, right? So like they're going to bring up something that works a certain way, and then now it's disrupted. What's going to happen? I think similar questions will come up about energy, right? So like, why are we seeing this? Why is the data doing this? What's going on? Energy is so important for living things, okay? So this is just a quick picture of it's looking at egg size and how many eggs there are, right? So if I have huge eggs, if I'm I think this is from fish, if I lay huge eggs, I don't lay that many, right? But if I lay a ton of eggs, look how small they are, right? And it's all about energy, because if I'm laying a large egg, it takes a ton of energy to do so. So I can only lay a couple. Where if I'm making smaller ones that are maybe going to be someone's snack, or they're going to be broken easily because they're small, well, I'm going to lay a whole bunch of them, right? And it ends up costing about the same energy. But again, there's a rhyme and a reason to it. Also, things that happen. There's things like examples of when organisms pause their reproductivity in terms of like they don't reproduce for a short amount of time in order to save energy. So in order to prepare for the long migration, certain monarchs will go on a reproductive diapause. So it just pauses their time of when they're going to reproduce to save energy, because reproduction, think about like if you've ever been around someone who's pregnant, it takes a lot of energy to do it, right? It takes a ton of energy. Your body changes immensely. An entire human being is formed inside a uterus, which is insane. So anyway, here's another picture of just relationship that we see, that the larger the body mass of an organism, the smaller their metabolic rate, right? So they don't want to be burning through their food because then they'd have to eat so much. They're already so big. Okay, so the larger your mass, the smaller your metabolism, because you would have to eat so much. So again, this is just something you don't need to know in terms of like specific information, but I wanted to show you a lot of graphical examples because you're going to for sure see graphs tomorrow or whenever you're taking the test. Another quick review, just making sure you know the difference between a heterotroph and an autotroph. So autotrophs are going to be down or they could be up. It depends on if your diagram is flipped. But if they are autotroph, troph meaning food, they can self-feed. So they are your photosynthetic organisms or your biosynthetic organisms. So either one, or chemisynthetic, where they're able to get energy from the from organic molecules. So but mostly just focus on photosynthesis here. So energy flows from the autotroph to the heterotroph. These are both examples of food chains. They're very linear. They're very not accurate in terms of what we really see. What we really see is these huge interwoven webs. Make sure with the arrows, the arrow goes to the mouth of the animal eating it. So just think like an arrow is like a fork right to the mouth, okay? So the arrow is going, it's showing energy flow. So this polar bear is eating a ringed seal, it's eating a harbor seal, and it's eating Arctic cod. Yum, chomp. Okay, that's where the arrows go, into the mouth. Also, make sure you don't always assume that the bottom is the producer. In this picture right here, your phytoplankton is the producer, but it's way up here. So a lot of kids will look down here and be like, what? Where's the plant? And I don't even know what a plankton is. But you do know that here it is and it's being eaten, right? And it doesn't eat anything. So if it's not eating anything, well, then it's an autotroph. Okay, going into population growth, because we only have about 17 minutes left. Reproduction without constraints is going to happen exponentially. This happens with bacteria a lot of the times. Eventually a restraint happens, right? They either run out of space or they run out of resources, right? So run out of food. So most of the time we see this logistic growth where carrying capacity is limiting that growth. And so they usually level off. In all reality, once a population reaches carrying capacity, it kind of like goes up and down, right? It's not this perfect line where carrying capacity is a steel roof preventing anything. But you'd see like up and down and up and down and up and down. But generally speaking, it stays pretty close to carrying capacity. There's factors that limit growth that are density dependent and density independent. Density dependent means that it's going to affect you more when you have high density, right? So like disease, predation, starvation, competition, waste, all these wildebeests. I have a thing about wildebeests, by the way. You guys should see this HHMI video on basically everything we know on ecology, from carrying capacity to predation to like the food web. It's from this scientist who lived in Africa and observed wildebeest populations for a long time. Anyway, so if you are living in high density populations, those factors are going to affect you more. Think of it like with humans, everybody. Like what's going on with COVID-19 right now? If you live in a big city or you live where there are a lot of other people, hospitals are busier, transmission rates are higher. If you live in the middle of nowhere, your density of population is low. You don't live by a lot of people. So I'm assuming if you're living with not a lot of people, we're not going to have a lot of transmission rates. density independent factors. They will affect the capita growth rate regardless of density. So those are things like natural disasters, right? It doesn't matter how many of you are living by each other, right? A fire is a fire, and it's going to get you rather, it doesn't matter how your density looks. Okay, so let's do a little bit of population density practice. So these are the equations you'll be looking at. And just in case you haven't seen them before, dy over dt is rate. Okay, d, I'm assuming means delta. So like if you remember from chemistry, delta, or in physics, right? Like slope is delta y over delta x, right? So that's the same thing as dy dx. It just means change in or rate. So we can figure out our change in population size over time. And to get that, you just do your births minus your deaths. If you're looking at exponential growth, you can look at your population growth is going to be based on this R max, which is the maximum per capita growth rate, times N, which is your original population size. So be prepared that you might see a question where they give you births and deaths, and they give you R max, and then they ask you for N. And that's okay, because you'll just first solve for your, do your births minus deaths to get dN over dt, and then you can plug in dN over dt here and solve for N. Right? So you could do N equals dN over dt divided by R max. So there you go. Okay, so here we go. In 1 year, a population of wildebeest, see, I told you I love wildebeest. I'm a weirdo. A population of wildebeest saw 250 new offspring, so those are your births, and 100 deaths. Oh, wait, why did I do that? I wanted to write the letter D. D. So calculate the growth rate. So all you're doing is births minus deaths. That's it. Okay? So 250 minus 100. Okay? So the growth rate is 150, new population, new members in a year. Now it says it originally contained 2,000 wildebeests. Okay? So that's your N. That's the population size. Now it wants you to calculate R max. Okay? So you plug in that 150 R max times N, which is 2,000. Okay, so you solve for R max. I know by now you all know this math, and I love it because you've corrected my math twice today. So if we do 150 divided by 2,000, we should get 0.075. I swear if my math is wrong here, I will be so sad and I will cry. I'm going to type it in my calculator again just in case. 0.075. Okay, good. Phew. Okay, now we've got a small town in Michigan. That's where I'm from, so I use that, and we've got lots of deer. They consistently counted 200 deer in 5.1 square miles, and it's consistently been over 100 deer since the surveying began. So they've been high in population. The carrying capacity is 15 deer per square mile. Okay, so K is 15 deer per square mile. Is the population above or below K? So I'm going to give you a second to think about this because this is not necessarily using any of these equations, and that's okay. You will be asked to do math problems that are just logical. They're just mathematical. It's not asking you to look at even a crazy equation. You're just doing the math. So give that a second and work on this problem. Okay, so hopefully you looked at the fact that we have 200 deer in 5.1 square miles. So all you have to do is 200 divided by 5.1, right, because it was saying per square mile, and see if that is over our 15 per square mile, which is the carrying capacity. So 200 divided by 5.1 gives us 39.2. So yeah, we are way over carrying capacity, which is a problem, right? Because then we've got all these community dynamics, where if you have one particular organism within the community that is too big, then they're going to start to influence the other organisms. And I have a picture of that in just a second. So with community ecology, I thought I had a picture, but I don't. But if we think about our, trophic pyramid here, and I could put, we'll do plankton, urchins, and sea otters, right? And this is true. Sea otters are a keystone species. If our phytoplankton for some reason were to decrease, the sea urchins would not fare well either, right? Because all of a sudden there wouldn't be many things for them to eat. If the sea urchins started to decrease, then the sea otters would also decrease, okay? Or if you get the phytoplanktons doing really well, well, then the urchins are going to do really well. And so then the sea otters are going to be able to have tons of food. So then the sea urchin population will eventually go down. And that's why when I was talking about once you reach carrying capacity, you do generally see this up and down motion, because there's a lot going on. There's a lot of things at stake, and it's not just predator-prey relationships. So real quick, the other relationships you're responsible for knowing is parasitism. So one is hurt, one is benefited. Mutualism, where both are benefited. And then commensalism, where one is benefited and then one, I just put 0 to mean like no effect. I would see this as a question where, you read like this ridiculously long paragraph, and then they ask you to, identify which is which. So just read carefully, underline, maybe write pluses and minuses, and just trust your gut. You guys know this. Okay, we're almost done. Last thing. Okay, species composition and diversity. So usually when I ask students about diversity, they go right to nucleotide sequences, and they think about like, oh, it's having a variety of, nucleotides, or it's even like a variety of alleles. If we zoom out, because we're in ecology now, we're looking at ecosystems, diversity has to do with species. Okay, so this is just a quick example. This is from these online biology simulations. I love them. If you were asked to do this equation, the diversity index, okay, first things first, the closer to 1 you are, the more diverse your community is. The reason why I don't use ecosystem, because ecosystem includes the non-living components, the abiotic components. And so here we're just looking at living things. So we're looking at a community, which is a variety of organisms. So your numbers are n, the total number of organisms, and lowercase n, and then uppercase n, total number of the species. Again, just like chi-square, they're not going to give you a chart like this, right? You're going to have to come up with this on your own. So I first like to, and again, they're not going to maybe give you this picture, but I like this picture as a nice example of what we're talking about. Realistically, I could see them telling you, hey, there's this population that has a diversity index of 0.62 and another one that has 0.31. Which one is more stable? And the higher to 1, the more stable the population is, or the community is. So anyway, I look here and I've got only one bird. So one bird, I've got 1, 2, 3, 4, 5 spiders. I've got 4 grasshoppers, and I've got 3, I wrote bird twice. Bunny is what I wanted to have here. 4 bunnies. Okay, so diversity not just has to do with how many different species, it's how abundant those species are. Like this bird is not very abundant, right? So anyways, you add these all up. So we have 14. Okay, so we have, 10, 14, so 14. So for each of these then, you're going to do little n over n squared. So 4 over 14 squared, 4 over 14 squared, 1 over 14 squared, so on and so forth. It's kind of similar to chi-square if you think about the math. 5 over 14 squared, yada, yada, yada. At the end, you need to make sure you do 1 minus all of these added up. Okay, so 1 minus all of these added up. All right. I'm not going to finish that because we are running out of time. delete. Okay. So when we think about stability, and I want to say that one more time, stability has to do with our community's ability to handle change, right? So here's my original population community I showed you. It wasn't very diverse. This one now, I've got butterflies, squirrels, beetles, a lot of each, right? This population community turned out to be closer to 1 than the previous. And if you think about that, they all have a role they play. They all have a niche they have. And if one starts to do poorly, there are others there to kind of take its place, and not in a bad way, but take its place as like, hey, we've got tons of producers. We've got enough primary consumers. We've got a certain amount of top predators. And so that one isn't particularly going to make the whole system collapse, unless we're talking about a keystone species. So the sea otters are a keystone species. That's why I drew that earlier. If a keystone species gets knocked out, then the entire ecosystem of that particular place could collapse. And so that's why a lot of ecologists, work with protecting keystone species. There's even this really ugly bird in California. I can't remember its name, but it's a keystone species. And so they're working really hard. Like every single living bird, it's a condor, it's a California condor. That's what it's called. So look it up. It's so gross, but I love it because it's super important. The California condor is so low in number that they have every single living one tagged. So they have a little GPS because they need to make sure that they're doing okay. And that they experienced an extreme bottleneck event, which made their genetic diversity very low. Like they're all very similar. It's almost like inbreeding. And so that makes them more susceptible to disease, less able to handle changes in the environment. And anyways, so if you're curious and it's not 10 p.m. where you live like it is for me, look up the California condor. It's a really good story and great review for the test. Anyways, so some scenarios that would influence change, changes to the environment, natural disasters, large scale changes. Like I said, if a keystone species were to change, that would influence everybody. Big key components here. The more diverse, the more resilient the community is to changes. And that's true with genetics as well. Remember, the more genetically diverse a population is, the better they are able to stay alive. So we only have 2 minutes left. So I want to end by showing you some error bars real quick. If you've never seen error bars before, we put them in using a statistical test called standard error of the mean. Okay, so this would be given to you on a test. I really don't think they'd ask you to calculate it. I mean, it technically is on your sheet, but basically it's the standard deviation divided by the square root of n or something like that. I don't have it memorized. But anyways, they'd give you the standard deviation. You'd be able to do it pretty easily. So anyway, we know mean means average. And, when you look at averages, you're like, wow, 30 to 69. And again, this is about like this animal that like shoots out poop so that it can stay safe. Anyways, you don't need to know what it's about. We're just talking about data here. So your averages here are 30 and 69, and your standard error of the mean, which is how much are your numbers varying from the mean? Is there a huge variation or a little variation? 3.1 is pretty good. But how you would find it is, and make your error bars, is you do plus and minus your mean. So I would do, like here, I did... Yeah, okay, so I did 30.3. So actually, I think I asked people to do plus or minus 2 standard error of the mean. So even though the standard error is 3, asking for double the standard error gives you 95% confidence. And when I mean confidence, it means that we can be 95% sure that what we are seeing is not due to chance. There's only a 5% chance that it's due to chance, which I know seems repetitive. But anyway, so that's where 24.1 and 36.8 come from, because I did plus or minus 6.2 for both of those. So when you graph it, even if it's a line graph, that's okay. You can do error bars on a line graph. This just happens to be a bar graph. I would add, I'd go up to 36 and I'd go down to 24. And then same thing here. I'd go up to 76 and down to 62. These error bars are not overlapping. when they do not overlap, then the differences we're seeing is legitimate, right? It's not due to chance, right? So here, you'd be able to reject your null hypothesis. You'd be able to say, hey, there is a relationship between these two things. This is statistically significant. You can reject the null. Okay, I know that was a lot. I appreciate you all for being here tonight. And, oh goodness, delete. If you live in the Eastern time zone like me, do not do anything else. Go to bed. You'll do fine. Wake up. Don't eat a huge breakfast if you don't normally eat a huge breakfast. Do what you normally do for food. Drink lots of water, and you're all going to do great, okay? Thank you so much, again, for being here tonight. And that's all I have. It's 10 p.m. You're all so wonderful. And good night. Thank you. We'll talk to you all soon. Good luck. I can't wait to hear how it went for everybody. All right. See ya.